Good morning, everyone, and welcome to Stanislaus State's 10th Annual Science Day. My name is Sunpreet Kaur. I am a STEM Outreach and Internship Coordinator here at Stanislaus State. I will be one of your co-hosts for today's activities.
Good morning, everyone, and happy Saturday. Hello, my name is Daisy Hernandez, and I am the CSU STEM Vista Outreach Coordinator here at Stanislaus State, and I will be your second co-host for today.
Stanislaus State is pleased to provide the community with opportunities to learn by engaging in live activities and recorded activities that will be shared today through Zoom and Science in Our Community YouTube channel.
Science Day would not be possible without dedication and commitment of Stanislaus State administrators, staff, faculty, students, and as well as campus members such as yourself. Stanislaus State is committed to student success. Our campus leadership strongly supports STEM educations and events such as this one.
I would now like to recognize the co-founders of Science in Our Community and Science Day, Dr. Mark Grosvenor and Miss Julie Fox. Without their guidance and commitment to the program, we wouldn't be able to have STEM programs like Science Day. Now, a quick message from Science in Our Community Director, Dr. Mark Grosvenor.
Faculty, staff, and students of California State University, Stanislaus, to the 10th Annual Science Day. Those of you who have participated in the past know that things are a little bit different this year. Due to COVID, we're having to do things virtually. Hopefully next year we'll be back in person, where we'll actually invite you to campus to tour our labs and do a bunch of really fun activities provided by the faculty, staff, and students of the campus. Do have to thank a few people. First, we have to thank our Dean of the College of Science, Dr. David Evans, for his support in our activities. We also like to reach out to the Dean of the College of Education, Kinesiology, and Social Work, Dr. Admin Mir, who also supports our activities, along with, of course, the faculty, staff, and students of the College of Science and other departments across campus that are also participating, many of which you'll see in the various activities we have provided for the day. I also need to thank the Office of Service Learning, the director, Julie Fox, and then Brett Foray, Brianna Hale, Sunpreet Kaur, and also the VISTA member from AmeriCorps, Daisy Hernandez, who also plays an important part in what you're going to experience today. So again, the faculty, the staff, and students are excited to be able to at least provide something to you this year. I know it's a shame we can't be on campus and have that opportunity, but please enjoy the activities we've outlined for you today, many of which will be available from here on out on our website. Do please follow us on social media as well, so that you can see what other fun activities we've got going on.
Thank you, Dr. Grosvenor, for this message. As a reminder, our first activity will be starting at 10:15. So please be patient with us. We're just allowing other guests to join us so we can start the activity right at 10:15. Thank you once again for everyone who's joining us today.
Yes, as Ms. Sunpreet Kaur said, the activity will start at 10:15 a.m. So take this moment as a coffee break or just to stretch your legs before we start our first presentation. Thank you. Welcome back, everyone. Now it is my pleasure to introduce our first live activity of the day, the science behind sugar consumption in our Western diets, with Laura and Gerardo. Please give them a warm welcome.
All right. Hello, good morning, everyone. So if you give us a second, we'll get started. Alrighty, so good morning, everyone. My name is Laura Plasencia, and I am here with
Gerardo.
And today we will be presenting on the science behind sugar consumption in our Western diet. All right, so we're going to get started with the concept of epigenetics. So epigenetics is the study of how our environment and behaviors can affect how genes are expressed. So genes, or short sections of DNA, are expressed when they are read and transcribed into RNA and later translated into molecules or proteins, which determine a cell's characteristics and function. So what epigenetic changes actually do is that they can interfere or boost with the transcription of specific genes and cause genes to activate or deactivate. So epigenetic changes are natural occurrences that can be greatly influenced by the environment, by age, disease state, or lifestyle choices. An example of this out in nature would be with reptiles and their incubation process. So usually temperature will determine the sex of the embryo when they're in cold temperatures or hot temperatures. It'll have an effect. So some other lifestyle or environmental factors that can have an effect on us would be chemical exposure, medications, or diet. And as you guys can assume by the title, today we will be focusing on the effects that diet has. All right, so here we have a processed food scale. So on the far left, we have our unprocessed food, which would be as it's found out in nature, and it's generally the highest in nutrients and in fiber. Then we have our processed, which is food that is canned or food that is frozen. It will usually still contain essential nutrients, but it will also have added sugars, added salts, fats, and in addition to that, it also has preservatives and added colors to ensure that its color remains throughout its shelf life. And lastly, we have the ultra-processed. So this will be most of our packaged foods, and they usually contain few to little nutrients or fiber. They're usually sourced from corn, soy, or wheat, and just have added fats onto them. All right, so we're gonna get started with this quick quiz by the Health magazine called "Are You Eating Too Much Sugar?" So we're gonna go ahead and put it on the chat. If you guys wanna go ahead and click on the link and do the quiz, I'm sorry. So it's just a few questions. It might either give you your results right away, or it will ask you to provide an email to where it will send you your results. Let's see.
Do we want to wait a little longer on the other page, or want me to move forward?
We can wait a little longer.
Well, definitely.
Yeah. So I'm going to go ahead and actually click on the quiz, and I'm just going to read out some of the questions. So do you drink your coffee or tea with sugar? And your options are always, sometimes, or never. Then we have, aside from coffee, how often do you drink beverages sweetened with added sugar, such as sweet tea, soda, lemonade, fruit punch, sports, drinks, or energy drinks? So that'd be once a day or more, a few times a week, or less than once a week. Then we have, how often does your breakfast contain a sweet item such as a donut, pastry, muffin, scone, sweetened hot or cold cereal, granola bar, or etc.? So this, we have most days, a few times a week, or less than once a week. The next question is, how often do you eat a sweet treat or dessert such as cookies or other baked goods, such as cakes, cupcakes, pies, brownies, ice cream, or candy? So again, most days, a few times a week, less than once a week. Then we have, if you buy yogurt, milk, or plant-based milk, such as almond milk, how often do you buy the sweetened version, such as vanilla yogurt, rather than plain chocolate or your flavored milk and pre-sweetened plant milk? If you drink alcohol, how often do you use a sweetened mixer, such as cola, ginger ale, tonic, sour mix, etc.? Always, sometimes, or never. How often do you eat condiments that contain sugar, including ketchup, barbecue sauce, salad dressing, and tomato sauce? That's most days, a few times a week, less than once a week. And then how often do you eat fast food? Most days, a few times a week, less than once a week. So hopefully you guys are able to get your results right away or through an email. But hopefully those questions were able to give you an opportunity to reflect a little bit on whether we're eating a lot of sugar. And I'll go ahead and pass it on to Gerardo, who will continue with carbohydrates.
Alrighty, guys. So I'm going to give you a brief explanation upon carbohydrates. So carbohydrates are the sugars, starches, and the fibers that are found in food. Carbohydrates are classified as simple or complex carbs. Simple just means that they are 1 or 2 sugar molecules in length, while complex means that they are 2 to 3 sugar molecules in length. So here below in this table, I have the most common carbohydrates. So pretty much sugar is a simple carb, but starches and fibers, they're both complex carbs. The difference is that starches are linked via alpha linkages. This means that they could be broken down easily by the enzymes in our gut, versus fibers, they are linked via beta bonds, and our gut cannot break these down. So that's what makes starches and fibers different. So now I'm going to show you guys a pretty cool video about carbohydrates. Hope you guys enjoy it.
Which of these has the least carbohydrates? This roll of bread, this bowl of rice, or this can of soda? It's a trick question. Although they may differ in fats, vitamins, and other nutritional content, when it comes to carbs, they're pretty much the same. So what exactly does that mean for your diet? First of all, carbohydrate is the nutritional category for sugars and molecules that your body breaks down to make sugars. Carbohydrates can be simple or complex, depending on their structure. This is a simple sugar, or monosaccharide. Glucose, fructose, and galactose are all simple sugars. Link 2 of them together, and you've got a disaccharide, lactose, maltose, or sucrose. Complex carbohydrates, on the other hand, have 3 or more simple sugars strung together. Complex carbohydrates with 3 to 10 linked sugars are oligosaccharides. Those with more than 10 are polysaccharides. During digestion, your body breaks down those complex carbohydrates into their monosaccharide building blocks, which your cells can use for energy. So when you eat any carbohydrate-rich food, the sugar level in your blood, normally about a teaspoon, goes up, but your digestive tract doesn't respond to all carbohydrates the same. Consider starch and fiber, both polysaccharides, both derived from plants, both composed of hundreds to thousands of monosaccharides joined together, but they're joined together differently, and that changes the effect they have on your body. In starches, which plants mostly store for energy in roots and seeds, glucose molecules are joined together by alpha linkages, most of which can be easily cleaved by enzymes in your digestive tract. But in fiber, the bonds between monosaccharide molecules are beta bonds, which your body can't break down. Fiber can also trap some starches, preventing them from being cleaved, resulting in something called resistant starch. So foods high in starch, like crackers and white bread, are digested easily, quickly releasing a whole bunch of glucose into your blood, exactly what would happen if you drank something high in glucose, like soda. These foods have a high glycemic index, the amount that a particular food raises the sugar level in your blood. Soda and white bread have a similar glycemic index because they have a similar effect on your blood sugar. But when you eat foods high in fiber, like vegetables, fruits, and whole grains, those indigestible beta bonds slow the release of glucose into the blood. Those foods have a lower glycemic index, and foods like eggs, cheese, and meats have the lowest glycemic index. When sugar moves from the digestive tract to the bloodstream, your body kicks into action to transfer it into your tissues, where it can be processed and used for energy. Insulin, a hormone synthesized in the pancreas, is one of the body's main tools for sugar management. When you eat and your blood sugar rises, insulin is secreted into the blood. It prompts your muscle and fat cells to let glucose in and jumpstarts the conversion of sugar to energy. The degree to which a unit of insulin lowers the blood sugar helps us understand something called insulin sensitivity. The more a given unit of insulin lowers blood sugar, the more sensitive you are to insulin. If insulin sensitivity goes down, that's known as insulin resistance. The pancreas still sends out insulin, but cells, especially muscle cells, are less and less responsive to it. So blood sugar fails to decrease, and blood insulin continues to rise. Chronically consuming a lot of carbohydrates may lead to insulin resistance, and many scientists believe that insulin resistance leads to a serious condition called metabolic syndrome. That involves a constellation of symptoms, including high blood sugar, increased waist circumference, and high blood pressure. It increases the risk of developing conditions like cardiovascular disease and type 2 diabetes. And its prevalence is rapidly increasing all over the world. As much as 32% of the population in the US has metabolic syndrome. So let's get back to your diet. Whether your food tastes sweet or not, sugar is sugar, and too many carbs can be a problem. So maybe you'll want to take a pass on that pasta, sushi roll, pita burrito, donut burger sandwich.
Sorry about that. My slide went out of control, but I hope you guys enjoyed that video. Now I'm going to explain a little bit of what sugar is. So sugar is a type of carbohydrate that helps provide energy to our cells. And the subtypes are monosaccharides, which are simple sugars, while disaccharides are two sugar molecules linked together. Sugar is derived in two ways, naturally occurring, which we get from fruits, honey, and vegetables, versus added sugars, which are high fructose corn syrup and table sugar. Table sugar is also pretty much naturally occurring, but I put it under added sugars because pretty much everything nowadays has added sugar. Like if we go to the store and try to buy oatmeal, we'll most likely find that the oatmeal has some type of sugar added into it. So I'm gonna go ahead and pass to the other side for my partner. Actually, I apologize. This is also my slide. So how many pounds of added sugar alone do you think an average American consumes in a year? 7 pounds, B, 22 pounds, C, 34 pounds, or D, 57 pounds? Are you guys able to vote by any chance, or does it not allow you guys to vote? Well, in case it doesn't, I'm gonna go ahead and move to the next slide and you'll find out the correct answer. So, oh, okay, you guys are, okay, nice, it did show. Awesome, okay. So let me go ahead and show you guys the answer now. So, there we go. So according to heart.org, an average American consumes about 57 pounds a year, or 71.14 grams a day of added sugar. So if we look at the recommended amount, we can see that we are over-consuming the adequate amount for added sugar. And according to this research, it also shows that most of our sugar is being consumed from snacks and sweets and from beverages, which, it does make sense. So now I'm gonna go ahead and move to the next slide, and my partner Laura is gonna explain it.
All right, so as you guys can see, the US total sugar consumption has been increasing since the 1800s. We began of about 2 pounds of sugar consumption per year. By the 1900s, that had moved to about 45 pounds of total sugars consumed per year. And now today, estimates range from 110 to 150 pounds of total sugars. So that would include sugars from our vegetables or fruits or even milk in the form of lactose. And it includes the added sugars that are added onto the sodas, the breads, and so many different other foods that we buy. All right, so where are people eating the most sugar and fat? So we've actually been seeing this Western pattern diet, which we see in developed countries and developing countries. And it includes high intakes of sugar, salts, meats, fats, refined grains, and just in general, more highly processed foods. And as you can see here in the US, we rank as #1 of the highest daily sugar consumption. And it's interesting to see that even within the top 5 ranks, we have Australia at the bottom of 95.6. So we're still about 30 grams, consuming 30 grams of sugar per day, more than even within the top 5. So we are consuming quite a big amount of sugar per day. All right, so we're going to go ahead and go through some interesting facts throughout some history about sugar. So in 1493, Christopher Columbus brought sugar plant seedlings, and that gave rise to the sugarcane industry. Then they began exploring with other types, retrieving sugar from other types of fruits and vegetables, and they began retrieving it from Beta vulgaris, or the sugar beet, which was cheaper to process, and that led to overproduction here in the Americas that made it affordable for all classes. Then in the 18th century, a French medical student identified the first patients of rheumatoid arthritis, which since then has been linked to sugar consumption having more effects on rheumatoid arthritis. In 1906, Dr. Alzheimer, a German physician, identified a form of dementia caused by inflammation in the brain due to high sugar consumption. This is still an area of research that they're still doing a lot of studies on to figure out exactly how it is that sugar has an impact on the brain and on Alzheimer's. But so far, they have found significant correlation to raise an alarm. And then in 1925, during the Prohibition, soda began to be dispensed in vending machines, and that led to like an increase of overall soda consumption. And by 1962, about 13% of American adults met the criteria for obesity.
And in 1967, a Japanese scientist was able to convert glucose into cornstarch. He was able to convert glucose and cornstarch to fructose. And due to that, in 1994, soft drink manufacturers switched to this cheaper high fructose corn syrup, which from here, then this led to 2021, 40% of adults are now considered obese. And if we flash back to 1962, only 13% of adults were obese. So yeah, that's not a good sign.
So a little bit more about high fructose corn syrup. So fructose is extracted from corn or beets or in sugarcane, and in the process, they remove a lot of the fiber and nutrients. So high fructose corn syrup, it's a cheaper way of creating sugar, and it has longer shelf life than other sweeteners, which is why most manufacturers decide to go with this type of sugar. It is found in 74% of packaged foods, and it can have from 42 to 55 fructose content in baked goods and beverages. It's also included in foods that we would consider healthy, like yogurt, energy bars, salad dressings, and canned fruit, and more. So high fructose corn syrup, along with artificial sweeteners, has been found to have an effect on our food reward system, which affects our overall appetite. So our food reward system is crucial to us feeling satisfied after eating, and it involves some of the same circuits as addictive behaviors, which actually explains why sometimes our appetite will increase and cravings will increase of sugary foods, especially the more high fructose corn syrup and sugary foods are consumed. All right, so too much fructose can actually turn into non-alcoholic fatty liver disease. So fructose is actually the only sugar that's processed in the liver. So as mentioned earlier, fructose can be found in fruits, like for example, in an apple. So usually when you would eat the apple, the fiber in the fruit would slow down the process in which the sugar would reach the bloodstream. But when we consume products with or food that has this high fructose corn syrup, that high concentration of sugar goes directly into the blood. And in this case, with fructose, it gets processed in the liver, which can become excess fat buildup in the liver. And usually this non-alcoholic fatty liver disease is when there's more than 5 to 10% of fat in the liver. This disease is the primary disease within liver. And then if it gets more severe, it can result in NASH, which is non-alcoholic steatohepatitis. And then if it gets even more severe, it can become cirrhosis, which is scarring of liver tissue, which blocks the blood flow through the liver, and it actually slows down the liver's ability to process nutrients and hormones, drugs, and other natural toxins. So the American Liver Foundation actually estimates that 80 to 100 million of Americans have this non-alcoholic fatty liver disease, which initially doesn't have too many symptoms until it becomes more severe. And another fact about this is that this liver disease is actually the most common in children liver diseases, and it has been shown to double its numbers in the last 20 years. In addition to that, this non-alcoholic fatty liver disease, it's one of the leading causes for liver transplants here in the U.S.
So overall, it's just good to have self-control over how much sugar we consume. If we don't put ourselves, we put ourselves at risk of becoming overweight and developing type 2 diabetes. And if a person develops type 2 diabetes, then there's more complications that can arise from there. That's why we don't got to go there. Therefore, it's really important to try to have some control of our sweets. And it is also good to try to stay within our body fat percentage range. And that could easily be obtained by a quick doctor's visit. So now that we talked about sugar, one switch. Let's see. Okay, it was supposed to you. Sorry.
Alrighty, so in this 2003-2004 study, they surveyed the nutrition of children and adolescents, and it was shocking to find that 40% of their food was coming from fatty food, and foods with a whole bunch of added sugars that offer little to no nutritional value. So that would include soda, fruit drinks, and grain desserts, which took up the majority of that 40%. And then it's another thing is that a lot of these, like the soda, the fruit drinks, also have artificial dyes, which I'll go ahead and explain further on the next slide. So food dyes are chemical substances added to foods and beverages to enhance the visual appeal. So there's naturally derived food dyes, which can come from fruits, vegetables, and insects as well, actually. And there are artificial dyes, which are usually preferred by manufacturers over the naturally occurring dyes because they provide more color options and more vibrant colors. So here in the US, 90% of the artificial dyes that we use are red 40, yellow 5, and yellow 6. These are very common within like Doritos and just a lot of the chips and snacks that more like the junk food type of snacks. These dyes are petroleum-derived substances that, although they're not deemed to pose significant health risks by the FDA, they are still banned in many countries. In addition to that, these specific dyes have been found to contain carcinogenic chemicals, but because they're in low concentrations, they are still allowed in the food. So something interesting to just keep an eye out is that the FDA does not require manufacturers to test dyes for neurotoxicity. Therefore, we can't really know the effects for like neurodevelopment when we're not testing for it. So some of the concerns with artificial dyes. So in 2008, the Center of Science and the Public Interest created a petition for the FDA to ban artificial dyes for their connection to behavioral problems in children. Unfortunately, they were not successful. But then since then, they have published new studies concluding that 9 of those FDA-approved artificial dyes, which are listed on the right, are likely to be causing hypersensitivity reactions, behavior problems, and they could be carcinogenic. So it's important to note that the studies that they conducted were done on mice and rats, which obviously we can't have a full comparison because they're different organisms, therefore behavior works different in different organisms. But the evidence that has come out through several studies, they do find it's a small, but it is a significant correlation between artificial dyes and hyperactivity. And it's still early on. We haven't done as much research to see the problems that it could have to like neurocognition. But that's as far as we found. Another thing is that sodium benzoate, which is a preservative, they have actually found conclusive evidence of that having an effect on kids who have ADHD and worsening their effects or from having ADHD. Another concern from these artificial dyes is that since 1955, American consumption of red 40, yellow 5, and yellow 6 have increased by 5 times. So in this chart, we see the ADHD throughout the years. So the National Health Interview Survey conducted a survey for 3 to 17-year-olds, and they assessed that from 1997 to about 2018, it has increased. It has had about like a 4 to 5% increase of ADHD. So we don't have the clear evidence, but there are some correlations that we see.
Alrighty, guys. So now I'm going to talk a little about our gut health. So our gut microbiome consists of 100 trillion microbial cells. It is considered a regulator of our overall health, pretty much because it has influence over our metabolism, nutrition, and immune health. And it also has a nervous system that helps produce hormones and neurotransmitters. The gut microbiome, pretty much another name for it would be natural flora, are microorganisms that protect the human body from pathogenic invaders. And they also help us with digestion and absorption of nutrients. And interesting thing too is that the hormone serotonin, the happy hormone, 95% of this hormone is produced in our gut by natural flora. I think that was pretty interesting. So now that you know that this hormone is produced in our gut, I'm going to explain how our gut and mental health are correlated with each other. So I don't know if any of you guys have ever experienced a nauseous feeling, anxiety, or tummy issues. Well, the brain and the gut are connected via vagus nerve, hormones, and natural flora. There's been actually a correlation between mental health problems and GI problems. Individuals who suffer from anxiety and depression, it is believed that they have disrupted natural flora due to the stress response. And research in animals have shown that animals with disrupted natural flora and inflammation of the gut have brain problems. And some of these problems that they have are Parkinson's disease, autism, anxiety, and depression. And, it's just pretty fascinating to know that, your gut has that much, impact over your mental health. So pretty much two things that you could get out from this slide would be, that it is important to know what we're consuming, therefore making sure our tummy feels good. But it also is, especially during these times, it is important to reduce our stress by finding something that we enjoy, not just to feel better, but also for our gut to help, gut health to be better.
And if I may interrupt, sorry about that. We're actually running low on time. So if we can just skip to that last slide, Gerardo, please. So we have just listed, one more. Right there, thank you. So these are just some ingredients and chemicals to watch out for. If you guys would like to just take a picture. The high fructose corn syrup, it does go by 61 different names. So it's just important to kind of try to keep an eye on the different names and try to recognize. Here's some of the most common artificial sweeteners, and again, the artificial dyes, if you guys want to get a picture of that. And thank you so much for listening.
Yeah, thank you guys.
We just wanted to get you guys to, or us to start thinking a little bit more about what we're putting into our bodies and the effects that that has. It's important that we focus onto eating for the nutritional value and not just for the flavor, even though that can definitely be hard sometimes. Thank you guys so much.
Well, thank you for joining us in our first activity of the day, and thank you to our wonderful presenter. It's such a well-written presentation and very interesting. So thank you so much. The next activity will begin at 10:50, so in approximately 3 minutes. So if you would like to go stretch, this is your time. And please don't forget to check out our recorded content as well. Thank you.
Welcome to our second live activity of the 10th Annual Science Day. The second activity is Glow in the Dark Cells, presented by Nora Shimon and Karina Guadalupe. Please help me welcome Nora and Karina.
Hello, everyone. My name's Karina.
And I'm Nora.
And
thank you for logging on with us. We're so excited to talk with you today about our glow-in-the-dark cells. We're going to go ahead and share our presentation with you and begin here. And so first off, I want to remind everyone, please utilize your chat and raise hand options. We welcome you to interrupt us at any point during the talk with questions or comments. And we're going to ask our moderators to help us keep track so we can get to you as soon as possible. So with that, let's get started. I'm Nora, and this is my colleague, Karina. And today we're going to tell you a little bit about our special cells that glow in the dark. So with that in mind, I'm going to hand it off to Karina.
thank you, Nora, and everyone. Thank you for joining us today. Although we would have liked to be in person, I'm glad that we're able to still share the science virtually. And so we're scientists in the Sukenik lab at UC Merced, and we'll talk a little bit about cells. Here you see 2 cells, and we've tricked them into making something called the fluorescent protein so that we can study how cells behave. And so to begin, I kind of want to talk about what cells are. Cells are the building blocks of life. As you can see here, they come in all sizes and shapes. And they all have very important functions. But one thing in common is they help make up everything in our body, from our lungs to our heart to even our brain. And I do want to point out that, we only see 5 examples here, but there are many different types of cells in our body. But these are just a few examples. And so we're going to be kind of trying to make this interactive. So again, as Nora mentioned, feel free to, type in the chat box. And so the first question is, what do we have in common with all of these 4 pictures? Can anybody take a guess? Let's see, let me open the chat here. Okay. Any guesses? Nope. Okay. Well, it can't be. Yes, they're
Great
all
guess.
living. Good job, Tammy. Well, these, we would need to put them in the dark to see if they glow in the dark, but yes, they're all living. So yeah, that was spot on. They're all made up of cells. All living things are made up of cells, and although they look very, very different, right? Plants, which cannot move, they depend on light to store their energy or make energy, to creatures in the ocean, that can swim and they're very thin, to something as big as a giraffe, or even something as cute and cuddly as this little kitten here. We're all made up of the same building blocks, cells. And so Nora here will talk to us a little bit about the size of cells and kind of help put it into perspective as to how small the cells are.
Yeah, so here we're going to think a little bit about size and scale. So like we mentioned, all these very different organisms have one thing in common, is that they're made up of these tiny, tiny units called cells. So let's think about size here. So here we have an example of a very tiny penny, and we have a grain of salt on the penny just between those little tiny pillars, if you can see there. So very, very tiny. So now we blow up this image of a grain of salt, right? And next to it, we have a red blood cell. And look how tiny it is compared to that. It's so small that you cannot see it with your naked eye. So the red blood cell is a very important cell. It's a cell that carries oxygen throughout our whole body, so our tissues stay alive and our muscles have energy. So it's very, very important, its function. And so being that it's so small and we can't see it with our eye, we need special equipment to study how these cells work. And Karina here is going to introduce that special piece of equipment.
Yeah, thank you, Nora. And so I do want to stress, for example, like the cell, the red blood cell, if we don't get proper distribution of oxygen throughout our body, for example, in our brain, it can cause us to have, for example, seizures or other diseases. So being able to have proper cell function is important, and being able to understand how they work is also important, because if they're not working, then we can find a way to fix it. And so we use a microscope to help us see the cells. Without any use of any tools, we can't see these cells. But with the microscope, it's able to magnify the image. And so it'll make it 1,000 times bigger. And we're able to facilitate how we see these cells. And so I'll give you guys kind of a virtual tour once we switch over to the camera. But I do want to say that although the cells are very small, it's very, very chaotic. It's very crazy inside the cell, similar to what you guys see in this picture, right? There's people crossing the street. There's a whole bunch of cars, taxis, and trailers going in different directions, even people on their bike here. And so it's very crazy. And unlike traffic, where it happens during traffic hours, certain times of the day, in the cell, it's happening all day, every day. So the cells need to have a way to stay organized. And the way that they do that is by having these machines inside the cell. And so some examples are, for example, the nucleus, which holds the DNA, and we'll kind of go into more detail into some of these when we're seeing the cell's life. But they just have a way to stay organized. Okay, and so let me go ahead and move to the next slide. And so although the scope helps us see the cells at a bigger image, for example, here, this is called a bright field image, where we can see the cells. It's still difficult to see the machines within the cell, right? And so in order to help us visualize or focus on the certain machines inside the cell, we use a glow-in-the-dark helper. A glow-in-the-dark helper, which are called fluorescent molecules or fluorescent proteins. And it's similar to, like, for example, when bikers wear this jacket at night and the light shines through them, they'll glow in the dark or glow at night. And so we do the same thing with our cells, where we make them glow in the dark and we're able to see the different components. And I'll go into a little bit more detail of like what this is. And so right now it's time to see some cells in action. And so we're gonna give you guys kind of a virtual tour first. So let me stop sharing so we can share our camera. And I don't know if you guys can pin us so that everyone can see it. I'll do like this. Right, and so this is our microscope. Here is the eyepiece, right? This is where we're gonna be looking through and seeing our samples. Our sample is gonna go, if you zoom in over here, this is the stage. And then these are the different objectives. And this is what we use to magnify them in different magnifications. And over here, this is the cameras. So these are the images that get sent to the computer, and what I'll be seeing through the eyepiece, you guys will be seeing on the computer. And so I guess let me just show you real quick. If you can point over here, Laura. I'm gonna show you guys the different light. And we're walking into our lab because we are going to be working with cancer cells. They're bone cancer cells, and so they're from humans. We need a separate room for the cells, and that's called the TC room. So if you guys follow me, we are making sure we're wearing our lab coats and our gloves, and we can go ahead and come on in. Even though we're in a separate room, we still need to use our safety cabinet to make sure that we keep everything inside. And so we are very clean to make sure that the cells stay in there. And because they are from the human body, they need to be at the same temperature as the human body. And so we keep them at the same body temperature. And if you guys see here, we have a lot of cell lines going on, but the ones that we're gonna look at are gonna be on this plate. So I'm gonna go ahead and grab them. Okay, and so now we're gonna go, I'll show you guys the play once we're in the TC room, in the scope room. All right. And so this is our lab. And so you guys come on in.
Thank you.
And so going back to the penny analogy, let me go ahead and grab the penny. Right, this is the size of the penny, and this in each little square that you see here, right? How many cells do you guys think that fit in each of these little squares? Can anybody take a guess? I don't know, I can't see the chat, but any guesses on how many cells fit in one square of these?
Ooh, good
Oh,
guesses.
wow, these are really, really good guesses. So 120. Yeah, so this one is actually from 100 to 500,000 cells per little well. And so, this, can hold a lot of cells because they're very small. And so now what we're going to do is we're going to put them on the stage. This is going to close down, but I'm going to take off the lid. And so now we're going to share the screen. So we're going to stop sharing the camera and share our computer screen for the microscope. And so we can see these, what I'm seeing on my computer, you guys will be able to see as well. Now, can you guys see the computer screen? Yes, okay, all right. And so, as I mentioned, right, this is a picture of the DIC. And so bright field, right, we're just looking at the cells, and it might be difficult to see, even though we have it magnified, right? So here, it can be kind of difficult. And the first thing that we're going to be seeing, actually, let me just share my screen. Just the screen. I can show this. And so the first thing that we're going to see here are going to be the nucleus. And so Nora's going to tell you a little bit about what the nucleus is. And go ahead, Nora.
Yeah, so the nucleus is kind of like the brain of the cell, where all the operations get their instructions. So inside the nucleus, DNA is housed, which is the blueprint for all of life. And some other things we'll find in the nucleus include proteins that have different pHs, RNA, which is what DNA is the template for, and lipids and minerals like fats and vitamins, and histone proteins, which are proteins that DNA wraps around so it can shrink down. Because if we stretched out all of the DNA in a single cell, it would be over 6 feet long. So they have to wrap around really tight around these histone proteins so they can shrink down and fit in the nucleus of the cell.
Yes, and it's important, right? Because I mentioned that the cell is very small and the nucleus is inside the cell. So it needs to fit in an even smaller area or space. And that's why the histones are important. And so we'll kind of show you guys here. This is what the cells look like without anything that, without any fluorescent molecules. But if we look at it, like, let's say we want to study the nucleus in this case, when we actually label it with the fluorescent molecule called DAPI, we see that we're able to see the nucleus. And if we actually look carefully over here, and if I switch between both, you can kind of see that where the overlap is, right? Where this right here, this is the nucleus, right? This one is the nucleus. This is the nucleus. And so labeling this helps us study the nucleus. And so let me open the chat. There's something in the chat.
Yeah.
Yes, that is very amazing that, a whole bunch of DNA can fit in that small space. Yes, good point. And so let me go back to the PowerPoint. And so the next thing that we're going to see is going to be tubulin. And Nora's going to tell you a little bit about the tubulin.
Yeah, so the tubulin is a really great structural support for the cell so it can stay nice and strong. It's like the cell skeleton. So we call it a cytoskeleton, like a miniature skeleton. So it makes the roads in the cell where things can travel across to perform the cell's function. So it's really important for support, and it acts as a road inside of the cell.
Yes, and so what you guys see here, for example, is, right, the nucleus is dark because that's not what we labeled. In this case, we're interested in looking at the tubulin, which helps make up the microtubules and the actin, which function as the roads. And going back to our analogy of the traffic, this is how things get transported from one factory or one machine to another machine. is that they can connect. And something that's really cool about this is that they can form and then fall apart and form something different. So they can form different lengths and at different sizes. And so it's very cool because, you know, this plays a part in like how the cell can change shape, and also affects like how they can respond to something by changing shape and being able to crawl around in different spaces. And so now I'm going to show you guys here on our computer. This is what the cells look like, as I mentioned, right? These are all different shapes and different sizes, right? And they look a little crazy, but these are the cells. And if we want to focus, for example, first on the nucleus, right, we're able to see it. And you can kind of see where they overlap, right? Where, especially in this one, you can see it overlap here. And when we look at the total cell, like you're able to see it still. And so what's different about the microtubules is we trick the cells into making this fluorescent molecule. And so not all the cells fall for this trick, and only some cells do. And so that's why for this next picture, you only see some cells that do start glowing in the dark. And if you can kind of see, if we zoom in, you can kind of see these little strings or these little spaghettis. These are actually the tubulin that we're looking at. And so right now we're looking at like all the roads of the cell. And as you can see, they can change. The roads don't always stay the same. In this cell, for example, the roads are more curvy and, you know, span over a longer area. And so, you know, this is how we're able to study, you know, the microtubules of the cell. And then the last thing before we kind of go into the cells, another thing is the protein activity, which is labeled by calcium. And so, Nora, do you want to?
Yeah, so calcium tells us how healthy a cell is. With our tagging it with this molecule, it'll turn brighter green if the cell is healthier. So this little stain that we use on the cell is a reporter of the cell's health. So it works by counting the number of reactions that happen in a cell. And by that, it glows brighter green and it tells us that the cell is healthier. But if it's darker, that means the cell may be dying or not so healthy.
Yes. And so this can be important because then we can study whether cells become healthier or unhealthy when we're adding, like, you know, different drugs to the cells and seeing how they react to it. And so here I'm going to show you guys again an example of the cells. Again, this one looks a little wonky. As you can already see, it's a different shape and a different size. But when we look at the actin, this is just looking if it's bright green or not. And in this case, you know, you can kind of see it's not too bright, but it's still bright green, meaning that, you know, the cell is still alive and it's functioning. Right. So that's how calcium works, but we'll do a little bit of some live cell perturbations, and you can see how the cell health will change based on the change of the calcium activity. And so let me go back to the presentation, and we're going to start doing some perturbations, but I do want to go into a little bit of, you know, a little bit of information. When cells, you know, are exposed to something that's not normal to what they're used to, right? They have various responses. They can either try and you know, initiate different signals and pathways to be able to respond and adapt to this change. But if it can't, the next step is just to die. It'll sacrifice itself, because if that cell is not functioning, and it will become sick. If that cell becomes sick, it'll get other cells sick. And one example of that is cancer, right? If cancer, what cancer is just the cells have lost the ability to sense when they're crowded. So normal cells, when they start growing, and they start growing to like a very large, like a large cells in a small area, right? If Nora and I are really like close to each other, we'll be like, okay, whoa, we're crowded, we need to stop. Well, these cells, these cancer cells, they do not have that ability. So they will just keep growing and growing and growing. And when they grow, they form these tumors, and that's what cancer is. And so, right, if and going back to our analogy, it's kind of like saying, for example, if the traffic lights stop working, then all the cars will just crash into each other. The people will also crash into each other. So it's very important for cells to know how to respond. And it's very important for us to understand how they respond. And so, right, we're gonna start by asking you guys, we have a couple of solutions here. And so we have some water. We also have some soap and some alcohol, as well as a whole bunch of salt. Salt or sugar, as if you guys were in the previous section, right? We can't eat too much sugary stuff, so we can also see how the cells react to that. So right now, I guess we'll open it up to the chat to see what do you guys want us to see on the first cell.
Do you want us to add water, salt, soap? You want us to add alcohol?
Some alcohol.
We have one for alcohol, one for water, one for soap. We have 2 soap. Let's add soap first.
Okay.
We're gonna bother ourselves by adding a lot of soap to them. So we're gonna see live how these cells react to this bothersome soap. Let's make some predictions. What do we think the cell is gonna do after we add some soap to it? Let's hear it in the chat what you guys predict.
Yeah. While you guys think of what you guys expect to see, I'm gonna go ahead and start focusing on the cells. So we can go ahead and watch this live.
So I'm going to show you Karina adjusting the cells so that we could see them nice on the computer.
Yes.
Right now, she's looking through the eyepiece and adjusting the stage so that the cells can be in a nice focused area for us to see. And this is really important when we're studying cells. We want to see them nice and clear. So Karina's a really good expert on imaging cells. And like she mentioned, the cells we're seeing on the screen now are cancer cells. So they're kind of out of whack. They're not regulating their behavior so well. So now we're going to watch what happens when we add soap to these cancer cells.
Yeah, so right now I'm just trying to focus on the cells. And then we're going to go into the calcium activity because that's what's going to monitor the cell health. Okay, and so these are a little bright, so I'm going to change the intensity a little bit so that we can see changes.
Yeah, so keep an eye. These cells are reacting. And we're going to see what happens when we add soap.
Okay. All right. Okay. So I'm going to turn off the light a little bit. I'm going to turn off one light just so we can see as much signal on the cameras and not get any background. So
yeah, our glow-in-the-dark helpers appear a lot brighter when there's no outside light. So that's why Karina's turning off the lights above us.
And then let me see. There's something in the chat. Oh, thank you.
Yes, great predictions. Great predictions. They might expand, they might die. So let's watch and see what happens.
Okay.
And so
right now, what we're looking at are the healthy cells. Okay, they look kind of like this. All right, and so now I'm gonna grab the soap. I don't know if you guys can even see me. This is the soap. I'm gonna open this, and we use a pipette kind of to add this. We'll see. We haven't tried this. Part of science is sometimes it works, sometimes it doesn't. Sometimes what we expect to happen won't happen, and sometimes it does. But this is the fun part of science, right? We are just exploring our curiosities and asking questions and trying to answer them. All right, and so here we go. I am about to add it. And let's see if we're adding something. Oh, it disappeared.
So keep an eye on the cells, everyone. They're going to be reacting to soap in a little bit. Karina's doing a little bit of mixing of the soap and cells. So sometimes the cells go a little lost. They go swimming in our little ocean of soap.
Oh, you see, so some of them just like, you see the brightness just totally disappear. So the cells that disappeared just completely died. And I'm trying to focus in and see.
Look at that.
Yeah, like some of these just completely died. These are still hanging in there. But if you notice here, right, this changed. And so that means that, you know, there's not much glow in the dark. And so this one you can kind of see, let me focus it. It might look different on your guys' screen.
Looks good.
Yeah.
So now we're seeing what soap does to these cells. So for anyone who predicted that it's going to kill some of the cells, you were right. And that's why it's really important to wash our hands. because the
Yeah.
soap kills the cells, and sometimes there are germs on our hands, and soap helps us kill those germs.
Yeah, and so now let me find, while you guys think, choose which other one we wanna add to our different cells. I'm gonna choose another well and see what that does to the cells.
So let's hear in the chat who wants us to add alcohol, who wants us to add salt, who wants us to add water.
And so right now.
Okay, I'm seeing a bunch of salt, so we're gonna add salt next.
Okay.
And see how the cells respond to adding a bunch of salt to them. Let's hear some predictions for what we think salt is gonna do to these cells. Let's think about how we feel when we eat a lot of salty popcorn or a lot of salty chips and we don't drink water. What happens to us?
Or like the previous talk where we eat a lot of sugary stuff.
Mm-hmm. Okay, we have a prediction for the cells might expand after they get lots of salt.
Right,
Great prediction. Ooh, dry up and shrivel. Another great guess. Oh yeah, that's very good. Thirsty cells. That's a great guess.
Some thirsty cells.
Oh, yeah.
So I'm gonna look at the DAPI first, and I am going to focus.
Okay, so while Karina's focusing on getting a nice image here, let's remember the DAPI is a nice little stain, like an ink, that stains the nucleus of the cell. Because like Karina was saying, there's so much going on in the cell. When we want to focus on one part, we use a little ink called a stain so that we can highlight that part of the cell. In this case, the nucleus. So after Karina adds salt, we're going to see what the cells do to respond to this pressure of salt.
All right, and here we go. I am about to add some salt, as you guys can see. If you guys can see, I know
Yeah, they
in the
can
dark.
see.
Okay, and so here we go. I'm about to add it. This might happen fast, right? Cells need to respond fast.
So I'm going to answer Tyler's question. These cells are bone cancer cells. So they're wonky in how they grow and grow and grow. They'll form tumors. Okay, so Karina's adding the salt. Let's pay attention to what happens to the cells. She's going to mix them, and we're going to see what happens.
I'm going to make these a little bit brighter, but you guys can kind of see that, the brightness is decreasing. It might be hard to look. Are you guys seeing?
Yeah, that's very clear.
Oh, you know what? I should have zoomed in into it.
It's okay.
Yeah, so I don't know if you guys were able to see it. It kind of happened like really fast, where these kind of shrunk. Can you guys?
So let's hear in the chat, whose prediction do we think was right? So the cells got dimmer, so that means some of them were dying because they got too stressed out. It was way too much salt.
Yeah. So let's try what happens to, if we want to look at the shape, let's look at one with the microtubules. So let me find one with microtubules, and we can see if we can actually see the structure of the cell change.
Yes. So let's remember what microtubules are. Like we mentioned before, the stain that we use to highlight the microtubules is called, Rita, what is that called when we stain the microtubules? Which stain is that for?
So we use actually a protein that binds to the microtubules, and this is called ensconsin. And so ensconsin recognizes what the microtubules look like, and it'll bind to it. Once it actually attaches to it, it'll emit that, it's bound to a fluorescent molecule, and it'll emit that green light that we see.
Right. So we're using a protein to help us find the microtubules in the cell. And once that protein goes into the cell and finds that microtubule, it's going to glow in the dark. So Karina's looking on all of our hundreds of thousands of cells, and she's trying to find one that is highlighting the microtubule.
Yes. So right now what we're seeing is the bright field, and this just helps us see what overall the cells look like. I did forget to mention, what you guys will be seeing is sometimes when the cells do die, and for example, like obviously I'm taking it out from the body temperature to the room temperature here. And so the cells are experiencing these changes. And I did forget to mention, when cells do die, we start to see called cell debris. And so we see some cells here, but when other cells die, right, we see this right here. Essentially what you're seeing are all the cell guts and insides. When cells undergo apoptosis, they start to like ball up, and maybe we'll see it with the ensconcing, but they start to form these little balls and fall apart. And so this is what is these are the remains of the cells. And if we look at with Daffy, right, it just looks like a whole bunch of stars, but these are actually just dead cells, dead cancer cells. So we don't feel that bad. And so, yeah, so that's, some of these might start to, apoptose and die like this. But let me go back to the live cells here.
So as we see, none of the cells are glowing in the dark with this particular light because it's just a regular light. So it's not making the cells excited, because when they get excited, that's when they glow in the dark. So she's looking around. We have hundreds of thousands of cells to look through. And Karina is our cell imaging expert. So she's looking through and she's going to find a nice cell for us to,
Nice and big,
yes, a nice and big cell. So now we're looking through. And Karina is looking in what's called a well, just like a well of water. We have a well of cells. It's super duper tiny, but in those tiny wells are smaller than a penny. There's 100,000 cells. So
moving to a different well.
Karina's moving on to a different well. So that means 200,000 cells that she's been looking through.
Right. And this one has more. So I do, when I add the different imaging solution, I do sometimes accidentally kill the cells, which is why some cells don't have that many. Some wells don't have that many cells. And so here, this one is great, right? You can see a whole bunch of cells. They have different lengths. And now let's see if they're actually expressing this fluorescent. Imaging.
Yeah, so like Karina mentioned, we trick these cells to glow in the dark for us to help us track the little factories inside the cells, the little tiny machines. Like we have organs in our body, so do cells. They're called organelles, which just means mini organs. So that's what we're doing right now. We're making those mini organs glow in the dark so we can study them.
And so here, this is a really good one, right? Like you can definitely see, these are the two nuclei. These are actually two cells that are probably dividing. And here you see all the microtubules, and you see that they look a lot different than, this cell, for example, and this cell, which is rounding up. And so now we're gonna look at this and see, what do you guys want to add to this one?
Yeah,
stop it.
Let's hear it in the chat. Do we want to add salt, alcohol, water? We got 3 for alcohol. Let's add alcohol to the cells.
We'll see why we shouldn't drink, at least at a young age.
Yes, Karina's right.
Drink here. This is alcohol. I've been adding like a lot of volume to the wells, but because alcohol is a little bit stronger, I'm gonna add a little bit less of what I've been adding.
Right.
Just so the cell won't completely die and like float away, because when they die, they don't stick to the bottom of the well. And so I'm gonna add a little bit less than that, and we'll see what happens. Any guesses?
Yes, let's make some predictions. How will alcohol affect these cells? And like we were talking about earlier, that rounded up little cell in the bottom left corner, that guy's probably not so happy. So he's probably not alive. But that bigger cell, that's glowing. We see the nice structures inside. That's probably alive. So we want to keep an eye on that one and see how that one's going to react to alcohol. Okay, Karina's getting started. Let's see.
All right, here we go. Let me know what you guys are seeing, because I'm not looking at the screen and I can't see it with my eyes.
Okay, Ariana says the alcohol should kill them. Great guess. Okay, guys, keep an eye on the cell.
You see those things floating around? That means that some cells are just dead and they're just floating around. And when I add more solution to this, right, they're just floating around. Let's see what is happening. Do you see anything getting brighter? Anything getting dimmer? Is the cell changing shape?
Ooh, it looks like it's getting dimmer. Kiana and Jennifer agree. It's getting dimmer.
Did this get brighter? I didn't pay attention, but let's see. Whoa, are the roads changing into a different direction?
Whoa, look at that, guys. Those are the microtubules. They're reacting.
You see how they're starting to not like unbind from here and moving to separate areas of the cell? Right? If I like get closer or further
Ooh.
apart, you see how this, you can kind of see them stretching out from here. If I move further from the cell, okay, maybe they're just a little drunk.
Yeah, the cell's not so happy with all this
But
alcohol.
If I do want to point out these cells over here, do you guys see these little dots these circles starting to form? These are called blebs. What the cell is doing is called blebbing. And so when the cell dies, in this case, this one is probably bigger, so it's taking a little bit longer, right? The microtubules, they need to send the signal from here all the way back to here or to different sides of the cell. So this is a lot bigger, but here the cells are blebbing. And so that means that they're dying, right? A chunk of the cell already came off and it's separating.
Yeah.
If I zoom in or out, right, you can see different blebs. And so this was probably the first one to come off. Over here, it's just, there's a big part that's just coming off. And so this cell is actually dying. I do want to point out that some cells, they do take longer to die. And so, it sometimes it's immediately right after that you can start seeing changes. Sometimes it takes hours. Other times, it just depends on how bad of a change that is. And so maybe I should have added more alcohol to see, but you can kind of see these cells that are starting to die. And so let's see. Oh, this one doesn't look that bad. Let me see. Let me move that way.
So what do we think? Do cells like alcohol? Let's hear it in the chat. I don't think so.
And then, yeah, look at this one. This one looks interesting. Sorry, now I'm just playing with the cells. I to play, but yeah, so you can kind of see that maybe this got a little bit brighter. But yeah, now the cell is starting to sense that there's alcohol, and it's like, okay, hold on, there's something going on here.
Yeah.
Right. And so now we're starting to see, and if I decrease the brightness, yep, it's starting to get, everything is starting to go over here. If you guys can see it here, it's a lot brighter.
Yeah, so let's remember, each of these cells, they started off alive. So when you're alive, you want to react to what's going on around you so you can stay alive, right? So some of these cells were overwhelmed when we added this alcohol. And so they, like Karina said, they started to bleb and form these little bubbles and fall apart and die. So they're not alive anymore, right?
So these are definitely not alive, and these are starting to die. And so now I kind of want to see, okay, now what do you guys want to see?
Yes, what should we add next? We have salt and we have water.
Yeah, so we added already to the other ones with calcine, we added salt and we added soap, but for these ensconcent ones, we haven't. So we can try adding it, but we also have water. What happens is, right, I don't know if you guys have heard of water poisoning.
Yes.
You have to drink like a lot of water, but it's possible that like if you do drink too much water, right, you have your salts in your body and your electrolytes that if there's an imbalance, then your cells will need to adapt. And so what happens is the cells will either swell up or they'll swell up to adapt to that. But if it's for longer terms, you can get water poisoning, and your body will react to that by saying, your cells will swell up and be like, hey, we have too much water in the body. And so what it will do is it will make you throw up the water. But yeah, so
that's how the body regulates itself when we have these stressors. So
Yeah.
like these cells are like balloons. If we put too much water, they're gonna burst. But like with salt, like we were guessing before, if they get too dehydrated, they're gonna shrink, shrivel up.
okay,
So they're not
and
gonna be
so
happy.
Let me find a different well, and then we can add something else. Let's see what it looks like here like this. So you see, maybe it's here, it's easier, right? You can kind of start seeing all these little gunky stuff. This is the blebbing cell. Maybe let me just move it a little bit to the side, right? All of these cells are actually blebbing. So they're dying. Oh, whoa, I should have imaged here. Look at this. So let me zoom in a little bit, right? Do you see all these little circles? These are all parts of the cell that already came off. And the cell, essentially the cell is dead already. And so you can see multiple cells are already having this reaction. This one hasn't gotten the memo yet, but all of these already have. And so what we're going to do is we're going to go into another well, and we can add something else. We still have water. We can add salt if you guys want.
So Kiana asked a great question. She asked, is this what happens to the brain cell after consuming alcohol? So what did we see? The cells were not happy and they shrunk a little. Is that right, Karina?
Yes, and some started dying. But so when we drink alcohol, right, the alcohol goes into our stomach and it gets digested. So the first ones to sense the alcohol are the cells in our mouth and the cells in our stomach. By the time it gets to our brain, it's not that, like, whatever gets extracted from the alcohol, it doesn't get to our brain. So those, to answer your question, in our brain, our cells do not do this. They have a safety mechanism, right? Because if cells in our brain started to die, then we would be in a very big problem, because the brain kind of controls everything, like our senses. It tells us when to breathe. So our body has these safety mechanisms to, protect our brain and other parts of our body, but especially our brain. But that was a very good question, Kiana.
Yeah, thank you, Kiana. So based off of Karina's fantastic explanation, let's hear in the chat, what do you think the two most important organs in the human body are? Kiana says brain, heart. Fantastic guesses. Ooh, lungs is another great one. What's that? The answer I think is correct is definitely the brain and the heart, because the brain, we absolutely need it. The brain never sleeps, because if it slept, then our heart wouldn't work when we were sleeping in the night, and our blood wouldn't flow, and we would be in really, really big trouble. So I think the brain is definitely our number one most important organ in the body, because it controls everything. And our heart circulates blood into all of our cells, all of our tissues, all of our organs. And remember, we showed that picture of the red blood cell? That cell is really important because it carries oxygen everywhere. And so the heart pumps around our red blood cells to all of our baby cells and gives it oxygen. And so that's really important, because if we didn't have oxygen in our cells, let's hear some guesses. What would happen if our cells did not have oxygen? If the heart stopped pumping around those red blood cells, and then the rest of our body did not have oxygen, they would be sad cells. They would die. That's very good. So our heart is really important to spread around oxygen in our whole body. So we'd be in big trouble without our brain or our heart working. So something that people do when there's an emergency and people aren't breathing, which means their cells aren't getting oxygen, is CPR. And so CPR is when someone else helps your heart beat and they push on it, help push your blood around your whole body so you can keep getting oxygen. And hopefully your heart starts working by itself later, so you'll be okay. So Karina's here looking at the cells, and she's adjusting how much light is going in and out of the cells, so we could get a nice clear image, so that when we do our experiment, we can see really clearly what's going on inside of the cells.
So I want to focus on this one, right? We can kind of see the microtubules, but we can also see the nucleus, where it's starting to have little dots inside. And so let's see what happens to this cell. We can kind of see both parts. All right, so I'm gonna stop this right here. What do you guys want me to add to this well? What have we not added?
I don't think we've added water yet. And to Jennifer's comment, we're so happy you're enjoying the presentation.
Oh. All right, so let me see. So do we say water or did we say, we already added salt to this one, right?
What do we say? Do we want to add water here?
Did we add salt already? No.
I think
So-
we did.
So I guess just a reminder, these are the ones we have.
Karina's going to show you our test tubes filled with different things. Some reminded us we have about 5 minutes left.
Oh, okay.
Okay,
Oh,
guys.
you guys can't see it, sorry, count.
They can see. Who says water? I think that might be a good one we haven't done yet. Okay, we have a couple votes for water. Let's go ahead and do
Okay.
that.
So this one's gonna be our last perturbation just because we are running low on time and I want to show you guys a cool little thing that you guys can try at home. Obviously not bone cancer cells, but something cool that you guys can play around with and get a feel of the lab. And so let me click live here. I'm gonna zoom in into this picture and focus it a little bit.
Okay, guys, let's make some predictions in the chat. What's going to happen? We add a lot of water to the cell. Remember, the cell's kind of like a balloon. So if we fill it too much, what do we think is going to happen? Pop,
Yes. So we'll
drown
see.
the cells. Let's drown the cells.
These cancer cells.
Oh, they're naughty cells. We
So-
don't mind.
I forgot to mention these cells. Let me add this first. So okay, let's pay close attention to see what happens on the screen. Gonna add. Is anything happening? Are they swelling up?
Great guesses in the chat, everyone. Let's look at the cells. Is it tough?
Is anything happening? Do you see any changes?
Subtle so far. Maybe we mix. See what happens when we drown this cell.
Let me add more water because I was adding the same amount of that I did of alcohol. So
Yes, so Karina's adjusting the volume because the alcohol was really, really strong. She only added a little bit to the cell, and that was more than enough to get a reaction. But with the water, the cells are a little bit more tough. So we want to add even more. So let's pay attention to the cell and see how it reacts to lots of water. Ooh, do you see something happening? The color's changing.
and so I do want to mention that like maybe right the water levels in our body are always changing.
Look at that.
And so our cells really need to be really good at sensing that change, right? Because if too many cells die, then that's a problem because then we're not having enough cells in our body. And as we mentioned, they help make up everything from our organs, all of our organs in our body. And so, right, when we drink water, when we sweat, when we cry, we're, losing water. So all of these changes of water in our body, our cells have mastered, right? So once they say, oh, we don't have enough water, they will send a signal and they will make you feel thirsty so you can drink water, right? And then, so that's what happens. Like, for example, you go on a hike, right? And you start to sweat, right? You're losing water. But what happens when you're sweating? You also get thirsty. And so your body is able to, for a bit, handle these water levels, these changes in the water levels and adapt. And so maybe that's why we're not seeing anything here. But
there's a bit of changes there.
you can kind of see the nucleus kind of got a little bit bigger, but and the little dots that we were seeing in the middle kind of just disappeared or got like more bigger here. So maybe that's one of the defense mechanisms that the cells have. Again, this is just more of like, I wonder why it does that. And so, for example, here it's like, why did it do that? And then we can start asking different questions and looking at the tools that we have in the lab and see how else can we study what we're seeing here. And so, it's really cool to see different things, but let's see if any of the other cells had any. Okay, yeah, so actually we're running really long time. So let me just stop here. I really hope you guys enjoyed it, but let me go back and kind of show you guys these different things. This link that I'll share in the chat, it's really cool. I'm gonna share it in the chat. Let me see if you guys get it.
So
Okay.
Karina's going to share a nice little activity that you can all do at home, where you can see how the microscope works
And-
virtually.
Yes, and so this is a different scope, but you can still go into learn and see how all the different things. But you can actually also look at some samples. And if you go to explore, You can also choose different things, right? There's animal slides, you can look at a spider leg.
Ooh.
And you can actually play around like if you were to focus it. Like it's really fun. You can try different things. You can even break it and it'll tell you broke it. And with that, we would like to thank you guys for coming to our session. This is our lab. This is our contact information. And if you want to check out all the research that we do, this is our lab website. And again, feel free to reach out if you guys have any questions. Thank you so much.
Thank you so much for joining us today, and we hope you enjoyed that. Feel free to use that link if you want to play with your own microscope.
Thank you for joining us for this activity, and thank you to our wonderful presenters. Our next activity will begin at 11:45. And also don't forget to check out our pre-recorded contact that we have on our YouTube. So thank you once again.
Hello, all new attendees and returning attendees. We are excited you to join us in the Google Trivia game with Eric Greenberg. Please give him a warm welcome.
Hello, how's everybody doing out there? Today's a beautiful day for science. And without some research, how could you even get some science done? So let's practice our Google skills and see what we can find out. Now, while this is called the Google Trivia Game, it's not associated or endorsed by Google or its affiliates. We can quite easily go ahead and use any search engine that we want. Now, there are many questions in life. So why not do an internet search for the answers? Well, mostly because of massive amounts of misinformation, but we can still use it for looking up answers to trivia questions. Now, you will need to use your web browser to do searches if you don't have a cell phone or tablet with you that you can do your searches on, and that's fine. Just double click the Zoom if you need to be able to access your web browser. Let's play the Google Trivia Game, not associated or endorsed by Google or its affiliates. The rules are simple. 1, I ask a trivia question. 2, you do an internet search for the answer. 3, you post your answer. 4, I show you my answer when the time is up. And 5, if you're close, you get a point. Don't forget to write it down. When we're done, you check what your Google Foo belt color is. We'll need to do a few calculations depending on how long we actually have to play the Google Trivia Game. I would like to ask my brilliant assistants that in the event of any technical difficulties, please unmute yourselves and let me know. And at the time that we only have 1 minute remaining, please chime in and we will do our final question and jump to the end. With that in mind, are you ready? Are you set? Go! First question, let's start with science. How many kinds of penguins are there? I'm going to put 30 seconds on the clock. Go ahead and look for your answer and post it once you find it. Just the other day, I asked a penguin if he wanted to attend a formal party, seeing as he already had a tux. He asked if he could bring his wife. I said, you can. He said, no, she's a toucan. Overall, it was a fairly chilling conversation. We have 10 seconds remaining on the clock, so go ahead and start posting those answers as you find them. We'll compare them to what I found out in 3, 2, 1. The answer I found was there are 18 different species of penguins. Amazing. Let's go for science one more time. A bit trickier. Which weighs more, a pound of gold or a pound of feathers? With 30 seconds on the clock, let me state that an ounce of gold and an ounce of feathers weigh the same, but there's a trick when it comes to pounds. Go ahead and do your search, see what you can find out. We have plenty of time, so check that out and see whether a pound of gold or a pound of feathers weighs more. 10 seconds remain. Go ahead and search. Remember, if you don't have time to post it to the chat, that's fine. Just give yourself a point if you get it right. Did you get that a pound of feathers weighs more? A pound of feathers is weighed normally, and you have 16 ounces per pound, whereas a pound of gold is weighed by the Troy system, which only has 12 ounces per pound. Wild, right? Watch out, sometimes measurements are tricky. Let's take a film question. Which surrealist painter was asked to play the Emperor in Jodorowsky's adaptation of the book Dune? 30 seconds on the clock. Dune is a fairly popular science fiction series set in a far fantasy future, where in order to navigate the stars, a spice needs to be harvested from a desert planet where giant worms roam. Those worms create the spice, and as long as the spice flows through the system, people are able to flow through the stars. That's right, the Dune series is where this term, the spice must flow, originally came from. Did you find the name of the surrealist painter? That was Salvador Dali. Let's take a terminology question. What does CPU stand for? 30 seconds on the clock. The CPU is inside of every device that's allowing you to watch this trivia competition and play along at home. That's right, it's an integral part to any computer system. Go ahead and pop that into your favorite search engine. Once you find the answer, go ahead and post that into the chat. If you don't have time, that's fine. Just mark down when you get an answer right. Here we go in 3, 2, 1. What does CPU stand for? Central Processing Unit. Okay, let's take another question. What is the world's smallest mammal? The smallest mammal in the world? I think it's pretty cute and a little surprising. Go ahead and head over to your favorite search engine, see what you can find out. There are a lot of tiny critters out there. Some of them are very small. You'd be surprised at how small a mammal can get. You'd be surprised at how big a mammal can get. Once this game's over, why not do a little bit of searching and find out what the various sizes are for mammals? It is surprising. Let's get those answers up. Did you find that the world's smallest mammal is the bumblebee bat? That's what I found. I thought it was pretty cute. On science, how many different color receptors, that is cones, do most humans have in their eyes? 30 seconds on the clock. Keep in mind that there are people who are colorblind and do not have as many color receptors, and there are even people who have more color receptors in their eyes. We're looking for what's considered the standard number of color receptors. Go ahead and do that Google search. We're just looking for cones, not rods, a specific type of cell that is reactive to different wavelengths of light. Did you find your answer? 3, 2, 1. Did you find that most people have 3, one for red, one for blue, and one for green wavelengths? Remember, some people have as few as 1 or as many as 4 different kinds. Now then, on pop culture, how many acting credits did Sir Christopher Lee have? With 30 seconds on the clock, go ahead and head over to your favorite search engine and see what you can find. Christopher Lee showed up in a surprising number of films. In fact, the answer might be very surprising. He had a long and varied career. He's shown up in a number of classics, and in fact, you've probably seen him in 1 or 2 yourself. Once you've found that answer, don't forget to post it to the chat, but if you don't have time, that's fine too. You're just going to want to take note and give yourself a point if you found out that Christopher Lee had 265 acting credits. Wow. Let's do another science question. How many kinds of teeth does an adult human have? Now, keep in mind, that's the kind of teeth and not the number of teeth. Humans could have a various number of teeth, depending on whether or not they remember to brush and floss. Remember, kids, don't forget to brush your teeth daily, and don't forget to floss, or your dentist will get a little annoyed. Now then, once you find the number of different kinds of teeth, go ahead and punch that into the chat so that you can share your answer with everyone else. Compare it. See what everyone found in 3, 2, 1. Did you discover that there are 4 different kinds of teeth, the incisor, canine, premolar, and molars, in an adult human? That's what I found with my search. Let's take a look at pop culture. Which band asked for a bowl of M&Ms with all the brown ones removed at every concert? With 30 seconds on the clock, go ahead and do a quick search for this one. Interestingly enough, they did this as a safety precaution. This band used pyrotechnics at the time, and after a stage collapse, they realized not everybody was reading the manual thoroughly that was included in the contract. To ensure that people were reading the manual, they had a strange request for snacks, a bowl of M&Ms with no brown ones in it. If they entered their green room where they'd take a break and saw brown M&Ms, they'd double check the stage. Did you find out that that band was Van Halen? If so, give yourself a point. Moving on to another science question. How many drops of water do you need to have a number of molecules equal to all the number of stars in the known universe? With 30 seconds on the clock, let me provide a little more information for you. A molecule is the smallest amount of something you can have and still be that something. In other words, water, in this case. If you had an entire drop of water, there would be quite a few molecules in it. There are a lot of stars in the sky, so how many drops of water would you need? And count out all those molecules to equal about the same number of stars in the known universe. Do you have your answer? Did you find out that it was about 10 drops of water? There are a lot of molecules in a little bit of water. Pretty amazing. Moving on to pop culture. Who invented the game Simon? With 30 seconds on the clock, you may not have played the game Simon, but your parents probably have. It's a popular game involving 4 colors and sounds that creates a pattern. After it plays a pattern, you play that pattern back to it using the buttons provided, and then it plays it back a little bit faster with an extra color and sound associated. This was a very popular game back in the day, and some food franchises would offer prizes if you could beat the game. Did you find out that the inventor of Simon was Ralph Baer? If so, give yourself a point. Moving on to a science question, who discovered the number E? Now, if you're just starting out in mathematics, the concept of a number that's a letter might be a little bit confusing, but don't worry, you'll get used to it. Some numbers are very complicated and very long. As a result, you don't really want to try to write them out every time you talk about it. Instead, you just use a symbol, such as a letter, to represent it instead. And one of those, a very common one, is the number E. Go ahead and see who discovered the number E and post your answer in 3, 2, 1. Did you get Leonard Euler? If so, give yourself a point. And let's check out television. Which cartoon has more than 630 episodes, but fewer than 1,000? Surprisingly enough, there are a few cartoons out there with over 1,000 episodes, but we're not looking for that. There are also quite a few cartoons out there with fewer than 600 episodes, but we're not looking for that either. With about 15 seconds on the clock, we're looking for a cartoon that has between 630 and 1,000 episodes. Now, this one might take a little bit of searching, but you've got time. In fact, you've got 3, 2, 1. Did you find out that the answer is The Simpsons? If so, give yourself a point. And let's ask you another science question. Now, what is the 400th decimal of pi? You've got 30 seconds. Keep in mind, I'm not asking for the 400th number of pi. I'm asking for the 400th decimal. Like I said, there are a lot of very large numbers out there, and that's why we use symbols such as pi, E, and others in order to know what we're talking about without having to write it all out, or it would take us a very, very, very long time. Speaking of time, right now we have about 3 seconds left on the clock. Go ahead and post your answer. Did you get 4 as the 400th decimal number of pi? If so, give yourself a point. If you got 3, you might have found the 400th number of pi. It will be a one-off. Moving on to pop culture. What podcast features a fictional radio station that broadcasts its local weather, news, announcements from the sheriff's secret police, mysterious lights in the night sky, dark hooded figures with unknowable powers, and cultural events? Podcasts are an incredible way to listen to audio. Using your favorite podcatcher, such as iTunes, you can subscribe to many free podcasts, which in turn have a number of different genres available, including science fiction, fiction, trivia, documentaries, pretty much everything under the sun. Think of it as a sort of modern day radio. Have you found the answer? If so, go ahead and post that. And in 3, 2, 1, did you find Welcome to Night Vale? If so, give yourself a point. Let's move on to question 14, and we'll do another pop culture. What is the airspeed velocity of an unladen European swallow? With 30 seconds on the clock, go ahead and see if you can find it. Now, keep in mind, this is a European and not African swallow. This question came from a rather famous Monty Python movie, Monty Python and the Holy Grail. In it, throughout the film, people would ask the question, what is the airspeed velocity of an unladen swallow? The secret was to ask back, did you mean European or African? Well, in this case, we mean European. Go ahead and see if you can find it and post it. Did you get about 24 miles per hour or 11 meters per second? If so, give yourself a point, and let's move on to the next question. In television, in the 1980s, the Tonka Toy Company sued a cartoon out of existence. What was the name of this cartoon? Funny story, the cartoon had nothing to do with any Tonka toy lines. It simply had a name similar to that of Tonka. Since the cartoon was not going to be renewed anyway, they decided to simply avoid the lawsuit by not seeking a new broadcaster to air their show. With that, Tonka had accidentally sued the cartoon out of business. Whoops, sometimes these things happen. Did you post your answer? In 3, 2, 1, did you get the Mighty Orbots? If so, give yourself another point, and we'll move on to another terminology. What does the word bit stand for? This term is used a lot in computer science and becomes fairly important. Go ahead and do a quick search for it and find out what it stands for. Now, it's a bit tricky too, if you'll pardon the joke, because it doesn't necessarily have 3 words. Strange, but hey, that's computer science for you. We make do with what we've got. Are you ready? Did you find your answer? Go ahead and post that to the chat. And in 3, 2, 1, if you got binary digit, go ahead and give yourself another point as we move on to our next question, history. Before a typo, what was Google's name supposed to be? Now, keep in mind, Google went through a few different iterations of names, but this was the name that they had intended prior to an accidental typo on Google. So, as you might guess, this may sound an awful lot like their final name that's used today, but it is not spelled the same. So, go ahead and do a quick search, see if you can find out what Google's name was supposed to be prior to the typo. Did you find your answer? Go ahead and post that in 3, 2, 1. As I said, it's Google, but it's spelled Googol. Wild, right? Moving on to television. A video game character had a PBS game show that aired more than 290 episodes. What was the name of this show? Pretty wild. I remember when I was a kid, there were a number of attempts to make television series out of video games with more or less success, but this one with 290 episodes was absolutely the most successful. It raised a number of children learning some pretty incredible facts, and we'll talk about those in just a second. And by just a second, I mean in just 5 seconds. Have you found the answer? Go ahead and post the name of that TV show. Did you get Where in the World is Carmen Sandiego? A game about geography when geography was changing so rapidly, they would have to redo their maps overnight. Amazing. Moving on to terminology one more time. What does IT stand for? 30 seconds on the clock. Keep in mind, I'm not asking for it, which is generally a reference to the scary Stephen King clown. I'm asking for the term IT. Now, you might have heard the term IT before, and certainly your parents have. It's a place that you go to when you have questions about various computers, phones, and other digital devices. So what does IT stand for? Once you find your answer, go ahead and post that to the chat. We have 3, 2, 1 seconds remaining. Did you find the answer was information technology? If so, give yourself another point. You're doing really well. Let's move on to another terminology. This one's kind of fun. Bite, nibble, chomps are all what? That's right. These words are serious words in my field of expertise. Go ahead and do a quick search and see what you can find. Making me a little bit hungry. I hope that everybody's ready for a nice snack once the Google trivia game is done. And if so, perhaps you can take a bite, a nibble, or a chomp out of that. And here we go in 5, 4, 3, 2, 1. Post your answer. Did you get a unit of information or a number of bits? That's right. Bytes, nibbles, and chomps are all ways of measuring numbers of bits. Crazy. Moving on to film, in the 1984 movie Ghostbusters, Dr. Venkman, played by Bill Murray, used his own variation of Zener cards to test students for psychic powers. What are these cards called? Hint, they are now a game. This one's a bit tricky, and if they hadn't been turned into a game, finding the answer would probably take a few minutes. But now that they're available for purchase, you can find the answer in about 30 seconds. Once you found that answer, go ahead and post it right to the chat. Zener cards were a real device used as people were trying to determine whether or not other people had psychic powers. The result was a resounding no. However, the game is quite fun. Once you know the name, post it up. Did you get the Anos Superior Quality ESP test cards, also known as Dr. Venkman's Anos Superior Quality ESP test cards? If so, go ahead and give yourself another point, and congratulations as we move on to the next question. Let's go for history. What was Google's original prototype name? Unlike their misspelled name of Google, which came from Google, they had another name. This name was used during the prototyping process. Go ahead and do a search. I think it's kind of a strange one, and I'm pretty sure you'll agree. Once you find that, go ahead and post that up. Did you find the answer? If so, go ahead and post it. We've got 5 seconds left on the clock. That's 3, 2, 1. Did you find that the answer was Backrub? This was followed by Googleplex, but Backrub is the answer we're asking for. I'm glad we don't have to do a Backrub search. That would be a little awkward. Moving on to the next question, let's do a pop culture. Duck dweeb, duck, 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 dweeb, dow. Ben Upland invented this famous blue superhero. Go ahead and do a search. It recently had a revival on a short-lived series on Amazon Prime. You might have watched it. If so, you'll know the answer. And if not, go ahead and do that search. You have 15 seconds on the clock. And hopefully you're finding your way through the myriad of blue superheroes, including the Blue Beetle, who we're not looking for. Keep posting those answers. And if you got it, did you get the Tick? If so, give yourself a point and congratulations. We're moving on to the next question. Terminology. What's that dangly thing in the back of your throat called? We've all seen it when we brush our teeth. Whoops, but it looks like Google Trivia itself wanted to answer. We'll skip on to the next question. That one was a freebie for the uvula. Whoa. All right, everyone, give yourself a point. Sometimes the answer's are right in front of your face. In this case, it was in your face. Moving on to television. In Star Trek, The Next Generation, what were those handheld scanner devices called and what were their main functions? Those are in the name. Now, go ahead and do a Google search for those devices. Now, keep in mind, there were a number of different Star Trek series, and in fact, a couple of them are airing now on streaming services. But we're looking for Star Trek, The Next Generation. They used these handheld devices for a number of different occasions, and they had a special variety for the doctors that had a detachable piece for close-up scans. Once you found the name, and once you know the main function, go ahead and post that. Did you get a tricorder? Its three main functions being sensing, recording, and computing? If so, give yourself a point and congratulations. A tricorder is shockingly similar to cell phones. Soon enough, once we have a few more sensing options, we'll really be there. The future's today. Wild. Back to science. Can bees recognize individual humans? Let's see if you can find the answer. Bees are often studied by a number of different scientific groups to see how they work together, communicate, and understand the world around them. Asking such vital questions as, can they recognize people? Do they suffer jet lag? And why do they make beep sounds when they butt heads together? Well, these questions and more are answered, and you can find them doing a simple internet search. And once you find that answer, go ahead and post it. And did you find out if bees can recognize individual humans? The answer is yes. Yes, they can. Go ahead and give yourself another point. Let's move on to pop culture. What does the 446th Pokemon evolve into? I'm going to give everyone 30 seconds on the clock because this one is a bit of a trick question. Make sure that you search it and find out. Once you know which Pokemon it is, if you're like me and enjoy the cartoon show, you'll probably know exactly what it evolves into. In fact, this is one of my favorite Pokemon. Go ahead and do a search. Once you find that answer, post that up right into the chat. We have 5 seconds remaining. 4, 3, 2, 1. Did you discover that the 446th Pokemon evolves into the 143rd Pokemon? That's right, the 446th Pokemon Munchlax evolves into the 143rd Pokemon Snorlax. Wild. Moving back to science, this sequence appears in pine cones. What is the name of this sequence? 30 seconds on the clock. This actually came up in a Tom Hanks film not too long ago, adapted from the book The Da Vinci Code. Go ahead and see what that sequence is and post it to the chat. It's a fairly interesting sequence, and it's used a lot, not only in mathematics, but also in graphic design. Once you have that answer, go ahead and post it to the chat. You've got 5 seconds remaining. 4, 3, 2, 1. Whether or not you posted it, if the answer was the Fibonacci sequence, go ahead and give yourself a point as we move on to the next question. Television. What was the trademark weapon in Xena, Warrior Princess called? Xena Warrior Princess was a spin-off of the TV series Hercules, which was based very loosely off the Greek mythology Hercules. In Xena Warrior Princess, they took a lot more liberty with the fantasy elements involved and gave Xena herself a special weapon that she could throw around to defeat evil enemies and monsters, and in some cases, cut down obstacles in her path. This weapon was had quite the impact on her enemies. Did you find out that the name of that weapon was the chakram? If so, give yourself a point, and we'll move on to the next question. Science! What is the name of the third highest mountain on Earth? We're not looking for the highest or even the second highest. We're after the third mountain on Earth. And bonus points to you if you discover where that mountain is located as well. But the third highest mountain is still quite large. I would not recommend just heading out in the middle of the day for a quick afternoon jaunt. You'd need to prepare for quite the hike. Physical activity is important in this day and age because you don't want to just sit in front of a computer all the time. That would be boring. So if you want to, go ahead and head over to the third highest mountain on Earth, Kachachunga, which I'm sure I mispronounced. Give yourself a point if you have the same answer as I do, or if you think that I'm completely wrong and your answer is correct. Moving on to the next question, we have film. What is the name of the 1990s movie that featured an entire city falling asleep and being experimented on by monsters? This is quite a scary film, so you might want to wait till you're a little bit older to watch it. However, it is a tribute to science fiction television series such as The Twilight Zone and Outer Limits. This movie is for people who are fans of such films or such television shows. I myself am quite the fan, and this remains one of my favorite movies, with a line I quote constantly: "Turn it off. Turn it all off forever." I say that frequently to my computer. Did you get the answer? Dark City? If so, give yourself another point. Well done. Let's move on. We're at about 30 questions, and we're back to pop culture. In Peter Aspich's original Evil Overlord list, how many rules were there? Now, the Evil Overlord list was quite popular about 25 years ago. This was a list of rules of what not to do if you became an evil overlord. Everybody's dream job, right? Well, until the hero comes and blows up your Death Star, or takes your crown, or destroys your magical crystal, or disassembles your ray gun. So many things seem to happen to evil overlords. Why not just join the hero side? But if you end up on the evil overlord side, you'll want to follow these 100 rules. The original list was a top 100. If you got that answer, go ahead and give yourself a point. Moving on to another science question. What kind of food is a kiwi fruit? This answer, is apparently ready to go. It was a berry. Wow, sometimes Google Trivia is a little fast on the trigger. Let's go ahead and move on to a similar question with science. What type of food is a strawberry? Give yourself a point for that last one, whether or not you succeeded the search, because the answer, juicy as it was, had shown right up. Now we're going to be looking for what type of food is a strawberry. Now, this one is a little surprising because foods aren't named after necessarily what they are. They're named after how they're prepared by chefs, which means there are a considerable number of foods out there that names aren't deceiving. That's why you want to know the scientific field name if you're going into a scientific field. That way you understand how they work. On the other hand, if you just want to eat them, why not just stick with their normal name? And you can pair this strawberry, even though it's a fruit, exactly like a berry. If you got a fruit for strawberry, give yourself another point, and we'll move on to the next question. What kind of food is a banana? That's right. We have just a couple more food questions here, and this is one of them. It's a little afternoon, and I guess I'm feeling a bit peckish for lunch. Once you find out the kind of food that a banana actually is, go ahead and post that to the chat and see if your compatriots have a similar answer. If so, high fives all around. If not, you might be using different search engines, and that's okay. Remember, a search engine is just good for initial research. After that, it's time to dig a little deeper, and you can find out that a banana is an herb. Crazy. Let's ask another food question here for science. What type of food is a coconut? Now, this one may surprise you too, because the answer isn't just one thing. Go ahead and start that search. What type of food is a coconut? Once you find that answer, go ahead and post that right to the chat so you can share with your fellow scientists in the making. You have 12 seconds left on the clock to find out what type of food is a coconut. Plenty of time. Remember, if you don't have time to post, that's fine. Just mark down if you got the answer in 3, 2, 1. Did you find out that a coconut is a fruit, a nut, and a seed? That's pretty nutty, right? Well, it's also a bit seedy, but Let's ask a film question. Which 1970s superhero movie had a motorcycle with a transparent shield? Now, superhero movies have actually been made for a very long time. A number of them, though, including this one, only aired on television. So despite being a movie, it is also just for TV. You can still find it some places hiding around on VHS tapes, an old way of recording television so you could watch it later. That's right. This 1970s superhero movie had a motorcycle with a transparent shield, not a windshield. So what was it? This 1970s movie was Captain America. Wild. Nowhere near as good as the modern day, still pretty good. Let's go ahead and move on to the next question, and we'll do another science. About how many bones does an adult human have compared to a baby human? The answer might surprise you. Let's go ahead and take a look. It looks like we've got about 1 minute remaining, so this will be our final question before we move on to discover what color your Google Fu belt is. Exciting. Go ahead and search, and once you have the answer, go ahead and post it. If you don't feel like posting it, that's fine too. Just keep in mind that you need to give yourself a point. Now, on that note, How many bones does an adult human have compared to a baby human? Far fewer. Adults have about 206 bones as compared to baby humans, which have 300 bones. Wow, the Google Trivia game is done. Keep in mind, Google Trivia is not associated or endorsed by Google or its affiliates. On that note, we're going to need to do a little bit of calculations because we weren't able to get through quite all of the questions. We did 36, so what color is your Google Foo belt? Well, still pretty close. Let's find out. Did you get between 0 and 7 questions? Well, not in this case. We're just going to do a quick calculation. And if you got 36 to 29 questions, you're a black belt. If you got between 29 and 22 questions, you're a red belt. If you got between 22 and 15 questions, you're a brown belt. And for purple belt, well, that'll be between 15 and 7. And if you got below 7, it looks like 4, you're a green belt. And if you got below that, you're a white belt in Google Foo. And you may want to do a few more searches in your spare time. Thank you so much and congratulations. Thanks for playing. I hope you have a wonderful day filled with science. Back to my brilliant takeaway.
Well, thank you, Eric, for this fun activity and just for all the amazing fun facts that you just taught us. So thank you, and I hope you all enjoyed it. The next activity will begin at 12:20, so please don't forget to check out our recorded content as well, and hope we see you soon. Thank you.
Hopefully you can see me.
Hello, all new attendees and returning attendees. We're looking forward to participating in the solar observing activity with you. Here is a warm welcome to our activity leader, Dr. Brian Mersoni.
Thanks. Hi, everyone. So man, take a breath. Hopefully this activity won't be quite as intense as the last one. But at any time, please put questions in chat. So I'm Dr. Brian Morsany. I'm a professor here in the physics department. So I teach physics and astronomy. And today, it's daytime, so we're not going to see any stars, except for the big one that's nearby. So we're going to look at the sun today. So we've got a couple of telescopes here. This is in the little dome that's north of Naragi Hall. We've got our big orange telescope here. It's like a 14-inch telescope, and it's set up to look at the sun in white light. So it's got a filter on the front that blocks almost all the light coming in so that we can look at the sun safely. Then this gold telescope on the side here, it's actually a special telescope made just to look at the sun. So it has a filter built into it that just lets through one wavelength of light. So it lets through a hydrogen line, so that we can see a little bit different view of the surface of the sun and see a bit more of the structure that's there. So let's see, let me bring up a slide just for a second, so that I can give you kind of an overview of what we're gonna look at. So this is what the sun looks like today. This is, a photo or a diagram from this morning. So there's a few sunspot groups on the sun that we're going to be able to take a look at. Hopefully we can see them. And this is coming from a site called spaceweather.com. So it's what I use. It gives you like this image. It'll kind of tell you about other things going on in space, asteroids, comets, all sorts of stuff. So let me see, since I've got the sun on my couple of cell phones here, let me see if I can bring up some images for you. We'll see if this works. I'm not 100% sure. I have to find Zoom on here again. Let's see if I can share my screen. Take just a second. And then go back to my camera. So in this telescope, it's pretty big, so we're just seeing a little corner of the sun. Let's see if I can steer it around a little bit. There we go. So if I go to kind of the edge of the sun, and then if I try to get it back in focus, so there's a couple of sunspots. You can just see them at the top of the screen there. That's that little group of sunspots, which I can't quite get into focus, but that isn't too bad. So they're right in the center of the screen now. There's also some dust on the lens. So, when I move it, the stuff that doesn't move is just dust on the eyepiece. That's the edge of the sun there. If I go back the other way, there's that group of sunspots again. And, they're pretty small compared to the size of the sun. So, if we go all the way across to the other edge, they're maybe only 1% or so of the diameter of the sun. But that means even those little sunspots, they're about the size of the Earth. They're still quite big. Let me see if I can switch to my other camera. And we can see a little more interesting view of the sun. I'll probably go back and forth a few times. Let's stop sharing on that one. Let's share that screen. It'll take just a sec. Okay, back to the camera. So here is a view of the sun. Let's see if I can center it a bit. There we go. Using this H-alpha filter. So here we're seeing the sun as kind of a red ball because it's just letting through this one wavelength of light where it's pretty red. If I zoom in a little, It's not going to show up super well on this image just because of my camera. You can see some patterns on the surface of the sun that we call granulation, where there's really convection happening. I'll show some video of it in just a bit. We can see, though, hopefully, some features on the edge of the sun. If I can try to get them in camera, yeah, right there. So right about here, a little bit lower, right about here, you can kind of see this plume coming up on the bottom of the sun right along, oh, you probably can't see that, right along the bottom here. And I'm going to try to get it to, let's see if I can play with the brightness any to try to get it to come up a little bit better. Boy, it doesn't quite want to cooperate. But if we go around the edge, let's see, you can kind of see, oops. When I don't get too much bloom from my camera, you can kind of see that feature coming up on the edge. So that's like what's called a prominence. So it is where you have, the sun has magnetic fields, you have like magnetic loops that are going over, and it has really hot plasma, maybe a couple 100,000 degrees that's trapped on that field line, and it's emitting, and you see this coming up. So it kind of looks like a volcano exploding on the sun. So yeah, you can sort of see the one there. Let's see if I can get any on, oops, now I lost the sun. Come back, sun, come back, sun. Let's make sure I didn't actually lose it. Oops. There's the sun again. That's actually not too bad. So there's our sun nice and centered. Probably not quite in focus. Let's see. And if we go around the edge just a little bit, trying to be delicate when I move this. Let's see, is there anything good on this side of the sun? Well, that's kind of in the lens flare for my camera. So let's head to the top. I don't think there were a whole lot of prominences here. And again, it's kind of limited by resolution of my camera. Aside from this one kind of nice one at the bottom right of the sun. So let's see. Yeah, only 100,000 degrees, pretty toasty. Yeah. Let's see. I think I'm going to switch back to my PowerPoint for just a minute. We'll come back and we can look at the sun some more. But I'll show some nice, like, high resolution videos coming from satellites in space that just stare at the sun all the time. So let me see if I can do that. Let's stop broadcasting from that one and then share my desktop again. So here's kind of in white light what the sun looks like today. Then I went and got some videos from the last couple of days. So here's the sun. This is taken from space, but again in kind of white light, with a filter. So this is showing the surface of the sun, what we call the photosphere. And if I play the video, you can see the last couple days. You can see the sun's rotating, so it takes about a month to rotate all the way around. This is that group of sunspots that we could see in the white light image. You can kind of see they're growing and changing a little bit. So this is the surface of the sun's about 6,000 degrees. So that's what we're looking at. But if we go to different filters, we can see different parts of the sun's atmosphere. So it's going to get warmer and warmer as we go up. What's the hottest part of the sun? So the hottest part of the atmosphere is going to be the outermost part, which is called the corona, and it's going to get up to a few million degrees. But the actual hottest part of the sun is going to be way down in the middle. So that's where nuclear reactions are happening. It's fusing hydrogen into helium, and there you're getting up to maybe 15 million degrees or so, and really, really dense. So let's see. So let me let you look at that video one more time. You can see the sun rotating nicely. If I go to my next slide here, this is showing a layer a little bit above that. So this is going to look more like what we were seeing in the H-alpha filter. We're seeing the next layer of the sun's atmosphere. It's called the chromosphere, a little bit hotter, still pretty dense. So now we're up to maybe 50,000 degrees. And here you're seeing those same regions where you had sunspots, but they're going to be much brighter. So they're hot. You have more energy. Compared to the Earth, how big are those sunspots? The ones we're seeing on the sun today aren't all that big. They're maybe, the size of the Earth, maybe a little bit smaller. So the sun's about 100 times as wide as the Earth is. When you get really big sunspots, you can maybe get things that are 10 times that big. If it was that big, you could see it by eye with a pair of eclipse glasses. Maybe, you get a couple of those every 10 years or so that are actually big enough you could see it without a telescope at all. With a filter. Don't stare at the sun with your eyes. That's a bad idea. I've actually, even had these where I was trying to use them as a filter for a telescope and melted them. So be careful. And you can even see here, if you notice where this got really bright all of a sudden. So that's actually a solar flare that's coming from that group where you had these magnetic fields release a whole bunch of energy and make a flare. And you can also see the prominences really nicely. Like here's a nice big one. I think this is the one we could sort of see through our telescope here on the bottom. So these again are these like magnetic fields holding up plasma that's radiating away. And you can see that they really change fairly quickly. This is over a few days. And it really looks like flames on the surface of the sun. Let's see, a little farther out. Oh, what causes solar flare? Good question. We aren't really 100% sure what causes those. I mean, that's still an area of research in solar physics, exactly how do they happen. What we do know is they're usually associated with sunspots, and we can look at how the magnetic fields are configured, and you can kind of think of magnetic fields act like rubber bands. If you stretch them, then you put more energy into them. So what you end up with is you have your sunspots, and often you have like a pair of them. You can even kind of see that here. You can sort of see these magnetic field lines connecting different parts of the sun. So they'll get stretched out. And then if you have something happen where you kind of cut that rubber band, all of a sudden it releases a whole bunch of energy. And that can give you this really bright flare. It can also eject mass that then is coming out of the sun. So there's always a solar wind that's coming out, but you can get these big eruptions happen when you have, so it's some sort of magnetic reconnection or magnetic fields reconfiguring, but not 100% sure the exact mechanism or exactly, okay, you see a sunspot and sometimes you get a solar flare or sometimes you get a bunch of them. Exactly why do you get a flare right now? We don't really know. So let's see, so this next video, again, same time period. This is looking a little further out, but this one really nicely shows some of these magnetic fields. You can really see like around here and here, these big loops of magnetic field, and they're bright because they have this hot gas that's trapped on them. If you ever played with like iron filings and a magnet, they'll kind of line up along in lines, look like those are tracing out the magnetic field lines. Same thing's happening here, but with this plasma. And there you saw the solar flare go off, got brighter all of a sudden. You can see this one boiling away on the side here with lots of little loops going up. Let's try it one more time. So as this one rotates off, you can see all these magnetic fields coming up above the surface of the sun. And then this was the sunspot group we were seeing. So, that sunspot was pretty small. The part that we see is dark, but this magnetic field active region is much bigger around it. And let's see, I think I've got one more video here, which is going out a little bit more. So here we're looking at the corona. So this is the hottest part of the solar wind. And now we're up to about 2 million degrees. And this is really kind of showing the solar wind flowing out, or how dense plasma is in this really hot region. So again, our sunspots that show up dark when we look in white light or in the optical look bright here because they have all this magnetic activity, this hot plasma trapped. You can also see where some regions it's dark. So those are actually places where the solar wind is coming out faster. And because it's coming out fast, the density is lower, more of it's going away quickly. So you actually have, if you have high-speed solar wind, it looks darker in this really high energy band. Is the sun solid? The sun's not really a solid. It's more like a liquid or a gas. So it's really made of like ionized gas, but it's really, really dense. So it's not like, it's not like air. The atmosphere we're seeing, well, this part of the atmosphere would be really low density, but maybe, maybe this one, the chromosphere, that's more like Earth's atmosphere. The sun, the average density, it's about the same as water. So the sun's just a little more dense than like ocean water or something. So you can kind of think of it as like being a big ball of water, but it's much hotter. So instead of being made of molecules, it's made of atoms, and those atoms are so hot that they're mostly ionized. So you have mostly hydrogen and helium, and then electrons floating around. So if something went and hit the sun, and it wasn't destroyed by the heat, it could sink down. It's not like rock that it would have to drill through or something. But you'd melt pretty fast because it's going to be hot. Maybe 6,000 degrees at the surface, and then gets hotter the further in you go. Let's see this one more time. Yeah, I can really see the solar flare in this one, right about there, coming from that active region. And you can definitely see the kind of fuzziness around the edges of the sun, where you have this solar wind, this plasma that's coming off into space. Is the sun perfectly round? Well, just like the Earth, it's not quite perfectly round because it is rotating a little bit. And so that'll tend to squish it a little because you're getting some like centrifugal force at the equator. So the gravity is a little less. So it'll kind of bulge out at the equator and get a little bit squished at the poles. But it's pretty close. I mean, it's, it only rotates every month. So it's almost perfectly circular. And the sun doesn't, like Earth, it doesn't have any mountains or that sort of thing on it. So the surface is pretty, it can adjust under gravity. You will have some irregularity on the surface because of these active regions and because of this convection that's happening. So, what you call the surface is a little vague because it's all just kind of gas that keeps getting denser and denser. But even like where you're seeing light from will be a little bit irregular. But yeah, it's pretty much the same shape as Earth, slightly flattened. Good, so I got about 10 more minutes. Let's see. Oh, and here's just an animation of that solar flare repeating again and again.
Did you get these from Space Weather?
Yeah, and so these came from Space Weather, and then this data is actually from the Solar Dynamics Observatory, which is a satellite in space, and it sits between the Earth and the Sun, so that it can stare at the Sun all the time. It doesn't have to keep going behind the Earth or anything. And let's see. I've got a few more minutes, but let me go back to the cameras for a bit. Let's just take another look at the Sun if I can. See if I can broadcast from my phone again. I might have to find the Sun because, of course, I haven't necessarily been paying attention here. Let's see. There it is. Okay. So there's the Sun in this nice H alpha filter again. Unfortunately, my phone camera, the resolution's not great. So it doesn't look quite as nice as it does by eye, and also the contrast isn't quite as good. But you can see this little prominence here. You see where it's a little bit extra fuzzy there. So there is like this big fountain of plasma, this like volcano happening on the edge. So if any of you were at the past couple of science days, you might have looked through one of these telescopes or maybe a smaller one we set up outside and taken a look at the Sun. And if you did, the last 2 years, it was probably pretty boring. You just saw a big white ball. There weren't really any sunspots. There weren't really any prominences on it. And that's because the Sun goes through like a cycle of activity about every 11 years. So it'll be really calm and inactive and, not very many sunspots, not very much flaring or anything. Then it kind of ramps up. And 5 years later, we'll have lots and lots of activity that'll last for a couple of years, sunspots all the time, flares all the time. Then it'll calm down again and then repeat that cycle every 11 years. So the last 2 years we were at this solar minimum. Hardly, the last 2 years there have been hardly any sunspots, hardly any solar flares or anything. It's finally starting to pick up again. So that's why we've got a few flares today and a few of these prominences. Another couple of years, we'll have lots of activity, lots more sunspots, maybe bigger sunspots, lots of this flaring. The atmosphere will be a little hotter. And then it'll go up and down again. And let's see, the last cycle, the last peak was around 2014 and 15, I think. And it actually wasn't all that great. It was kind of maybe only about half as many sunspots as previous cycles. So we don't really know what's going to happen with this one. The minimum was pretty long and deep, so it was really inactive for quite a while, for a couple of years. So who knows? We might have another kind of weak cycle, or maybe it'll ramp up and we'll have a more impressive one this time around. And how active the Sun is will also affect things on Earth. So the Sun's spitting out the solar wind and flares, and sometimes those flares will have big balls of plasma that come out and can hit the Earth. And fortunately, the Earth's mostly protected because we have our own magnetic field, and the solar winds magnetize, so they kind of repel each other. But some of that plasma will get captured, and then it makes the aurora on Earth, so the northern lights and southern lights. So in California, you probably don't see those. But if you're a bit further north, so if any of you, live in Washington or especially Alaska or somewhere, you can see those when you're at the solar maximum. When there's more sunspots and more flares, you're much more likely to see them. And especially further south, you'll get these big balls of plasma running over the Earth and making really bright aurora that move further south. So the last cycle wasn't that impressive. The one before that, though, so in 2000, 2001, my uncle lives up in Susanville in Northern California. Actually could see the aurora at least one time, where he had a big called geomagnetic storm. So you had a bright aurora that moved way far south. So it was actually down over like Oregon and Northern California. So if any of you live up that far north, you might have a chance to see some aurora in another, 3, 4, or 5 years. And space weather is a good place to actually see, are there predictions, has there been a CME, is something going to hit us, or coronal mass ejection, one of these big balls of plasma. Let's see. So any questions that have come up? I know I've been talking nonstop, so I have to give you a chance to interrupt.
The same things that cause sunspots, solar flares, or what causes sunspots?
Oh, okay, good question. That's actually perfect, because then I can switch back. And I think I have a slide for that. Stop that one. And let's share this screen again. So let's see. So here's just like some pictures of some sunspots. So if you get this really close-up view, high resolution, the sunspot is basically a place where you have really strong magnetic fields. So you can kind of see like around the edges, you'll sort of see this trapped plasma. And normally the sun is convecting along, so you have hot material come up and then it spreads out and cools off and falls back down. Same would happen like if you have a pan of oil on the stove. It's hot on the bottom, the hot oil rises, and then it'll cool on top and sink back down. Or like a lava lamp, that's convection. The stuff is, you have a lamp on the bottom, so it warms up, floats up, and then eventually sinks. But where you have this sunspot, these magnetic fields don't let the stuff sink back down because they're kind of holding it in place. It's ionized, so the magnetic fields trap it. So it'll actually cool off a little bit, and that's why these sunspots look dark. Instead of being 5,500 degrees where it's nice and bright, they'll maybe only be 4,000 degrees. So they'll be a lot, enough cooler that you can see the difference. They're still pretty warm. And then you'll often have these magnetic fields associated with sunspots. Sometimes you can even see this nice pair with these big arcing lines connected. And you could see that a bit in the videos of the sun. So then if you get these magnetic fields to get twisted in the right configuration, they can kind of break and give you a solar flare. Here's a nice big prominence. Let's see, did I have a solar flare video? Yeah. And then when that happens, you can get, so this is a really big flare from 15 years ago or so. It's a really nice big flare. It sends off this big eruption of plasma that gets shot out into space. Then it hits the Earth. That's that noise coming in. So that noise is the, you get a bunch of cosmic rays coming that are actually, in this case, hitting this satellite and kind of overwhelming the detector. And that happens in just a couple of minutes. But then this ball of plasma came and hit the Earth maybe 2-3 days later and causes aurora and lots of disturbance in the Earth's magnetic field. And I forget what exactly your question was.
What causes sunspots?
What causes sunspots? Yeah, so it's really where you have this like active magnetic field. You have this stronger, like twisted up magnetic field that's kind of holding everything in place. And I guess that magnetic field is kind of caused by the convection of the sun as well. So we don't really know why there's a solar cycle. People try to do simulations of the sun and figure out why do we have solar flares? Why do we have the sunspot number vary? Here's a drawing of it so you can see this pattern where every 11 years or so you get no sunspots, then a bunch, and then none, and then a bunch. But we don't really know why is this cycle every 11 years? Why is it not a different number? Why is there a cycle at all? Do other stars have solar cycles? We're not really sure. Okay, so I've got one more minute. So if anyone have any last question before I sign off? If not, thank you for being here. Hopefully next year will be in person and you can come look through some of these telescopes yourself, get a little better resolution view of it that I can give you on the camera. Thanks, everyone.
Thank you, Dr. Mersoni, for that wonderful presentation with all that solar information. Really great to have that.
You're welcome.
Thank you for joining us for this activity, everyone. The next activity will begin at 12:55. And also don't forget to check out the pre-recorded videos we have on our YouTube channel. Hello to all our new attendees and the returning attendees. We're excited to introduce Moises and Bartha from Warriors Chemistry Club as they will show you how to make homemade slime.
Can you guys see it? It's not on.
Yes.
I can get it to the slide, Moses. There you go.
There you go. Perfect. All right. Sorry about that, guys. Little technical difficulties. But hello, my name is Moises Nunez. I'm the president of the Warriors Chemistry Club. And today we're going to be making homemade slime.
So hello, thank you for being here today. My name is Berta Cervantes, and I'm the event coordinator for the Warriors Chemistry Club. And okay.
So next on up, we're going to look at the purpose, of the whole experiment. So I think the main thing is just making your homemade slime. It's your personalized slime. You can make it however you want, to your own preference. Actually looking at a few of the other slime kits I was looking at, it's actually cheaper using this recipe than buying, slime kits that from Amazon or Walmart that are like $20 to $30. It's a safe at home experiment, so you can do it anytime, at your house, and you don't have to worry about anything going on. And lastly, you get to learn about the chemistry behind the slime making. So it's a fun experiment, and there's science involved as well.
Just a little warning to parents: it is very safe, but if you're going to be using food coloring, it could get us slightly messy. So, just a little warning. So materials: contact lens solution, Elmer's glue, baking soda, a bowl, a spoon. Other materials: food coloring, glitter, scented oils if you want your glitter to smell good, and the mini styrofoams. And you could get all these materials at Walmart or the 99-cent store or Michael's. So basically what you do is you grab an empty bowl or container and you pour 4 to 6 ounces of glue. And if you want your slime to be stretchy, then you add 1/4 to 1/2 cup of water. added everything into your bowl, then this is the time when you could add 2 to 3 drops of food coloring. You could add glitter, you could add your oils, you could add your styrofoams or anything that you would want to use to decorate it and give it a little bit of color. After you have added the decorations, then it's the time to add 1/2 teaspoon of baking soda and you mix it thoroughly. And you may need to add more depending on the amount of glue. After the baking soda, that's when you add a tablespoon of contact lens solution and mix well. And so I tried it this morning, and depending on the amount of glue that you use, that's the amount of lens solution that you're gonna need. So what did we say, Moses? If you add 1 to 2 ounces?
Yes. So what happens is if you add like 1 to 2 ounces, you want to start off with about 1 tablespoon of contact lens solution, and you add it slowly. That way you make sure like it mixes well. If you add it too quickly, what can happen sometimes is that your slime will kind of become hardened and it won't really be stretchy. So you need to make sure like you add it gradually. Like once you add your contact solution, it's your slime activator. So once you add it to your solution, you just have to make sure that it's already forming. And if it's still a little sticky, you're gonna want to add a little bit more contact solution, but you would want to add it by increments of 1 tablespoon each time until you finally, got what you prefer of like, if it's too sticky or if you don't want it too sticky, if you want it, stretchier. So yeah, you just kind of go based off your own preference. So it will vary because it does depend on the amount of slime. I mean, on the amount of glue that you use.
So then when you have added everything, you just need the slime until everything is well incorporated and ready to play. And that's basically it. So following, Moses is gonna explain the science behind the slime.
All right, so I'm still mixing it around. I'm a little behind, but pretty much it's starting to look like this right now. I still need to add my contact lens solution, but I mixed in green and blue food coloring so I can get like a teal looking color. I don't know if you guys can see, but that's what I have so far. But the slime forms, it's a non-Newtonian fluid, and many of you may not be familiar on what this is, but if you've learned in school, you've seen that you learned about like solids, liquids, and gases. So a non-Newtonian fluid just means that it has mixture of properties of both solids and liquids. So that's why it appears like it's running, but at the same time it's still a solid, which is the slime. And the glue is actually made up of long chains of polyvinyl acetate molecules, PVA glue. So what happens is when you add your slime activator, which would be your contact lens solution right here, it forms like a chemical reaction with your glue as well as your baking soda. So when this happens, the slime activator comes in contact, chemical bonds are formed, and it'll kind of create like a cross linkage between both your glue and your borate ions from the contact lens solution. So yeah, it's a little complex. Like a lot of us you may not understand it once you get into high school or other chemistry, courses that you take further on in college. But that's pretty much what's happening with the process.
And this is a chemical reaction that occurs. The polynyl acetate reacts with the borate ion, and this is what we get right here.
So yeah, so like how I was explaining right now, this is the polyvinyl acetate alcohol. That's like pretty much the chemical structure of it on the top left corner. And then you have the borate ion. So once they come into contact with each other, there'll be a form of hydrogen bonding. Like I said, you may not be familiar with it, but pretty much there's just linkage between the borate ion and the polyvinyl acetate that forms the slime, and allows it for it to be stretchy and for you to play with. So is there any questions that you guys have? Do you guys get stuck on anything? I'm still working through it. Still forming right now.
So I did mine this morning. This is what it looks like.
Yeah, I did a demonstration earlier as well, and it looks something like this too. I believe I felt like I added a little bit too much contact lens solution. So like this is what happened if you add too much contact lens solution on there, or like, do you see how it's kind of hard to break apart like that? It loses like its stretching ability, like the flexibility. So you want to make sure when you do the procedure, it's not you don't add too much contact lens solution. So, okay, there's a few questions on here. All right, so let's go with the first one. What is your favorite thing to add to your slime? I mean, for me, I like a simple slime, just as long as I got the clear glue. You can also get glitter glue, whatever works for you, but I like the clear glue just so you can kind of see through. It's kind of cool in a sense. But I just like adding mainly just some food coloring. So like if you add, for example, I think the box right here, food coloring, it tells you which colors you desire. Like it says red, yellow, and then teal. So it kind of gives you like an estimate of what the mixture of colors can give you. So like I wanted to go for a teal, and I've got like this color of slime right here, teal color looking. And then, so let's say, okay. Yeah, it does kind of look like Play-Doh in a sense, especially if you get the white Elmer's glue. Let's see. So if you pour too much contact lens solution, it turns into a solid. So not necessarily, it won't turn into a solid, but it just makes it harder for you to stretch out. Like this is like the earlier slime I did earlier. And you kind of have to like pull it away slowly for it not to break. Because if you do it too rapidly, it'll snap when you add too much contact lens solution like that, as you guys can see. So it won't turn into a solid, but it just makes it, the flexibility of it, like the stretchiness decreases, like from my experience that I've done this. How long does it last? I'm honestly not sure. That's a good question. I think if you store it and you maintain it like in a concealed, container, you can have it you know, I'll probably say at least a few days, maybe up to a week. It just depends on the conditions that it's stored in. So let's see, great activities, seeing these reactions and information helps the student to start getting familiarized with this information. Yeah. Could you explain the chemical bonds more? Okay. Yeah, I could. Berta, did you want to share the slide again so I can kind of explain them what's going on? Or did you want me to share? Yeah. So, all right. So just a little like chemistry 101, you know, that you guys will get into once you get more into high school courses. So the chemical bonds that form, so pretty much, you know, all these products consist of different kinds of chemicals. You probably have already heard of, like, you know, the common ones, just as carbon, hydrogen, nitrogen, oxygen. You know, that's like the most common ones in the atmosphere that you probably heard of. So a lot of these compounds that we deal with, like in everyday life, they contain these, you know, different kinds of chemicals inside and different kinds of elements. So for me to kind of just simplify, you know, the reaction, because it's pretty complex in a sense. Like, you won't really learn this until you get into like college courses, or maybe in high school as well. But as you guys can see, the polyvinyl acetate, the alcohol right there, do you guys see how it has an OH? So the OH, just when you see that in a chemical compound or structure, it just signifies that it's most likely going to be. So that's what the OH signifies. And the O stands for oxygen and the H stands for hydrogen, right? And now the borate ion, yeah, and then one thing I forgot is that the lines chemistry, the lines right there, they demonstrate a little like a chemical bond between the carbon and the oxygen. So every line right there is just a chemical bond between both elements, right? So the borate ion is kind of similar in structure. It's also known as a boric acid, and as you can see, you know, it's similar in the sense that it has another OH in it, like another OH. So meaning signifying that it's an alcohol. So those are chemically bonded together. The OH is bonded together, and it bonds as well with the boron. And so yeah, it has in total has 4 OH groups, which, you know, signify an alcohol, right? So when you link both of these compounds together, like let's say you put them in a solution, and you mix them around, eventually those two compounds are going to form chemical bonds between each other. And as you can kind of see like the linkage on the right side, where there's a chemical bond from the oxygen of the borate ion with the hydrogen of the polyvinyl acetate. And that's what's called cross linkage, or in chemistry, it's called just hydrogen bonding between both, because I don't want to confuse you guys. It may seem like too complex, but the hydrogen contains, it's like pretty positive, while the oxygen is like a negative charge in a sense, or it's more electronegative. It's the right word to say, actually. So the oxygen is going to want to attract that positive, you know, charge of the hydrogen. So in a sense, it's forming some kind of bond between both of them, and that's what kind of creates your slime because of the linkage. So like at first it's just a liquid, but then once you mix in your slime activator, it turns into like this gooey, you know, slime pretty much. So that's mainly what's going on. Like, I mean, I hope I didn't lose you guys, but that's like the main thing that's happening right there. It's just this cross linkage between the polyvinyl acetate and the borate ion through the oxygen from the borate ion with, you know, part of the hydrogens from polyvinyl acetate. So they just form chemical bonds between each other. Because one's more negative and one's more positive. So when they come in contact, they're gonna want to link together. And you'll kind of see that more like once, I mean, this is just like a little introduction, but you'll see that more once you learn more about chemistry. That's just like a very simplified explanation of it. And I hope I didn't miss anything. Because like it makes sense, you know, it'll make sense for me, but for other people, I'd be like, if you've never seen it around, it's a big, you know, it's pretty complex. Is there any other ion or alcohol that can be used in place of the one in this current reaction? Okay, that's a great question, actually. So yes, definitely, you can use boric acid that's in contact lens solution, but you can also use different types of saline solutions. It doesn't necessarily need to be boric acid, but I think just because I think it's on the safer side of one of the alcohols, like, you know, there's less sensitivity when it comes in contact with your hands and stuff. You can probably use other forms of alcohols that can become slime activators. I know there's different processes that my little sister was explaining to me about. You can use, I believe you can use like borax powder. I know people that use shaving cream, as well as, what was the other? Oh, Tide, like, and detergents that contain the same kind of chemical compounds. So yeah, she was explaining it to me because she's more into slime making. But
so this one, I did it in the morning with borax powder. The only difference that I noticed is that it's even slimier. So this is borax.
Yeah. So yeah, you can look around. I think there's so many different recipes that you can use. But I just, when I did my research on looking into, you know, recipes that you can use for slime making, I know some people are sensitive to the borax powder. So, you know, if you happen to be, you know, sensitive to it, this is like a safer, easier way for you to, you know, go about it. And so like this is like my slime that I just made right now, the teal one. My little sister helped me kind of knead it together and put it together. But yeah, this is the final result, as you guys can see. So this one's pretty stretchy. I didn't add too much contact lens solution this time. So as you can compare from my other, from the results of my earlier one, you see this one's kind of just like, this is better like if you want to use it just to like play with just your hand, you know, maybe release some stress. But this one's just a lot more stretchier. You see the difference when you add a more slime activator. So that's pretty much what's going on. But yeah, do you guys have any other questions, any other explanations that you want me to further elaborate on? Could you explain more about covalent, polar, covalent, and ionic bonds? And which ones are used more in this reaction? Oh man, it seems like it's turning into a chemistry class. You know what? I have an idea. So yeah, it is a hydrogen bond that's occurring, kind of as I explained. But Dr. Alamão, would you mind explaining a little bit more to the audience? I don't know if you can, since he's. Okay. Yeah, he said that's fine. So I'll have actually one of the chemistry professors from San State, you know, to explain to you guys more in detail, because I feel like I just covered a simple steps. So I'll have him explain more to you guys since it's here.
Okay. All right. So I think, well, I mean, there are different type of reactions, and then at the end, you know, when you combine two compounds together, you form different type of interactions. In this specific case of the slime, he's showing the pictures that these two interactions taking place between that oxygen and the hydrogens, is what is called the hydrogen bonding in there. So if you compare in that pictures, you can see that they show the interaction with the dashed line. So it's because that chemical interaction is a little bit different than the one that I show with the bold line. So the bold lines are covalent bonds, so they are stronger. So when you have the dashed lines that are represented in the other pictures, those are the hydrogen bonding interactions, are a little bit weaker in there, but are strong enough, to keep the compounds together in there. And actually, that weakness that exists in there also gives you that flexibility, right? So that sliming behavior that you have is because it's not as strong. Because if you have something that is very, very, very strong, you're gonna have a solid, right? So that's what it was. And then you start to put more boric acid in there. What you keep doing is just packing more boric acid, and then more pack. You create something more and more rigid and more solid. So that's just in general what exactly is going on in terms of the interaction between these two different molecules in there. In terms of ionic bonds, there are no presents in here that I can see, but normally they happen when you have two elements that are ionized and they're just interacting because the difference in charges that exist between them. But essentially, I mean, once you perform chemical reactions, you're gonna have different type of interactions, but they are not all present at the same time. They are just specific for the type of reactions that you're doing. So in this case, it's just some hydrogen bonding interaction between borate ions and the polyvinyl acetates in there. So I think hopefully that helped. But you guys are doing great. I think my daughter is in the other room. She's making her, so she's stretching her all the way there.
Thank you so much. I appreciate the explanation, Dr. Alman. But yeah, so yeah, you guys can kind of see, what's going on. Dr. Alman was able to elaborate more on what I had said earlier. So thank you, Dr. Alman, for doing that. But yeah, it's definitely, chemistry is definitely really fun and interesting, and you'll learn more about this, the more classes that you take. I think for sure you'll take it in high school, I think junior year. So 11th grade, you'll take the class, and that's where you get a more understanding of what's going on, and you'll understand, maybe some of the things that we couldn't cover today just because we're on a limited amount of time. But I hope you guys were able to, learn some things. If you guys, if you guys like chemistry, I definitely encourage you guys to look into it. I mean, and I know some of you guys are still a little too early, but it's definitely a great major to go into, and we have a lot of fun. We do a bunch of experiments and other outreach opportunities, so I think you guys will like it. But I don't know if you guys any more questions that you guys had, or
So we have one comment. Chemistry is fun and everywhere. Yes.
Definitely, yeah. Chemistry is, you can do a lot with chemistry. I know. I mean, it's fun, and you also learn, about biology through chemistry, and vice versa, and physics and everything. All sciences are interesting and fun, but I don't know. I like chemistry, and I'm pretty sure some of you do as well. But anyways, I think that's it for now, unless you guys have any other questions or comments that you guys wanted to add.
Thank you for being here today.
Yeah, thank you for coming by and allowing us to present. And hope you guys have a great one.
Well, thank you, Moises, Betha, and the Warrior Chemistry Club for doing this wonderful activity. I hope you all have fun making your slime and just being creative, adding those glitters and just all those fun little things. And now I would like to thank you all, the audience, for joining us on our 10th annual Science Day and also being our first virtual one. Hopefully next year we will be able to see you all in person and have our usual Science Day. But thank you so much for attending our first one.
And please feel free to check out the on-demand videos available on our YouTube channel. If you have missed any of the earlier live events, They have been recorded and we'll share them on the YouTube channel as well later during the coming week.
And please do not forget to let us know how you enjoy Science Day by completing the survey that you will all be receiving at the end of this webinar. And lastly, I wanted to thank the faculty, staff, and student volunteers who made Science Day happen. Without them, we wouldn't be here. So thank you so much for all your hard work and dedication. And have a great rest of your day.
Thank you, everyone.
Thank you.